Welcome, everybody. I'm delighted to welcome you to the Institute of Psychiatry, Psychology and Neurosciences Research Festival 2021, whose theme is Across the Lifespan. And we're celebrating the innovative and exciting research taking place at the IoPPN here at King's College London. Please note that we're recording this meeting, and anyone needs captions, they can press the closed caption button. And unfortunately, in light of the ongoing pandemic, this year's festival is taking place online rather than in the Wolfson Lecture Theatre at our campus in Denmark Hill, where I've always enjoyed listening to the presentations. This means we will, in a good way, be enjoyed by a far wider audience than usual. So I'd like to welcome you all, whether you know a lot about the IoPPN or whether you know a little. We're pleased that you're here to join us, and thank you for finding the time to join us. As you know, the study of the mind and the human brain is one of the most exciting and important areas of advancing medical science, and making a difference is the heart of what we do. We have an enormously rich history, pioneering research, enhancing patient care, influencing policy change, raising awareness of conditions affecting the brain, or sharing our knowledge through education. And for the next 3 hours, we'll be showcasing some of our amazingly talented and inspiring research and work, with 22 5-minute talks and 2 keynote talks. I'd also like to take a moment to thank all our funders and grant awarding bodies, many of whom are in the audience today, and thank you for finding the time to join us. This past year has been a hard year for all of us, but I'm delighted that you're all joining us today to celebrate what the IoPPN does best. I'm now going to hand over to Professor Meta, the chair of the Research Innovation Committee, whose committee organises and runs the festival. Mittel, over to you. Thank you.
Thank you, Ian, and welcome to the Research Festival. The Research Festival is organised by the Research and Innovation Committee, which is a committee run by the research staff at the IoPPN. What we do in the committee is we want to enhance our research competitiveness, we want to promote innovative working, we provide career development opportunities and training in many areas, and mentoring for our staff as well. We really value fostering of early career researchers, and we also administer some awards and prizes for excellence in research. And I'm very pleased today to announce the 4 awardees of our Early Career Research Awards from the autumn round. So they are, and you can see on the screen, Andrea Dupreez, Andrea Dupreez, who is given the Dean's Prize, the IoPPN Prize, Maria Navakarada, who also gets the IoPPN Prize as well. And we have 2 prizes which are funded by a Biomedical Research Centre, funded by the National Institute for Health Research, and that's Lucy Vaines and Grace Lavelle. So normally we would applaud these 4 great individuals and they would come up and receive their awards, but we congratulate them over the internet today. We have a new round coming up, and the closing date is the 15th of May. And for those staff in the IoPPN, the email circulars have gone out, but if you would like more information, please contact Annika Ancliffe or Matthew Kempton. I'm very pleased to announce a new researcher award, new for this year, which is the Open Research Award. And this is an award to recognise those who use open practices. This is practices to make their work more robust, more accessible, more transparent, reliable, and replicable as possible. Applications are now open, and they are open to students and staff at the IoPPN at any career stage. We have 2 awards. One is an Early Career Researcher Award for open research, and that is open to PhD students through to lecturers. And we also have a second award, which is for established researchers, or an entire research group can apply as well. Okay, so if you want more details, they will be available in the next circular about research funding, but you can also contact Annika Ancliffe directly. And we'll be live tweeting this event thanks to Mental Elf as well. And the hashtag you can see on the screen is the IoPPN Festival as well. Great. Well, thank you very much for joining us. I'm now going to hand over to Dr. Paolo De Luca, who is the Programme Chair for this year's festival, who will chair the first session. Thank you very much.
Thank you, Mithal, and thank you all for attending this first online version of the IoPPN Research Festival. And I'm Paolo De Luca, the Research and Innovation Committee Lead for the Research Festival. As Ian just said, we have an excellent lineup of speakers who will give a snapshot of their work on a wide range of topics across the lifespan, from pregnancy to old age. They will highlight the changing nature of mental health and mental disorders across people's life. We have 23 speakers, and aside from the keynote speaker who have a longer 20-minute slots, everybody else will present for 5 minutes maximum. Mithal and myself will chair this afternoon sessions, and we'll try to keep the event on time and remind people when they are over time if that needs be. There will be a break around 3:30, and we aim to finish the event with the closing remarks from Professor Matthew Grubb at 5. So before I start, I just have a couple of additional housekeeping announcements to make. If you have any question to the speaker, do use the chat function. Speaker will stay around for 10-15 minutes after their presentation to answer any questions. And if there are too many questions, we will focus on the most popular ones. So do upvote any question you think should be asked and answered. Also, a lot of hard work has gone into organizing this event, and I would like to thank the members of the Research Innovation Subcommittee and the Press and Communication Department Office and IT who have worked very hard in preparing this event. This is very much a team effort, so many thanks to Annika Ancliffe, Josephine Manford, Maria Magdalena Petrinovic, Jonathan Humor Hartig, Hannah Warren, Trevor Brooks, Amelia Remington, and Robin Magin. So now, without further ado, it is my pleasure to introduce the first speaker of today. She is also the first of our 2 keynote speakers, is Professor Grainne McAlonan, Professor in Translational Neuroscience. Over to you, Grainne.
Thanks very much for the kind introduction, and hopefully everyone can see the full screen version of my slides. So I would really like to thank the organisers for inviting me, and although the title of the talk is about autism, I hope that you'll find the content more broadly relevant, not only to other neurodevelopmental psychiatric conditions, but to all of us as individuals. And I'm going to start with the problem for autism research. So although there are well-reported group differences in measures such as executive function, social cognition, and brain imaging indices between people with and without an autism spectrum condition, they actually don't tell us very much. And here's a useful exercise which my colleague Eva Loth shared with me. As you can see here, 2 groups can be statistically different, but still overlap. So if you look at the effect sizes, or how large the differences between 2 populations are, to start to separate 2 groups, you need effect sizes of well over 1. And on the chart on the right, effect sizes reported in the autism literature hardly ever reach 1. So although there are statistical group differences between people with and without autism, mostly the populations overlap. And autism is really not a discrete category. And so why might this be? Most of you will appreciate that autism spectrum conditions are really very diverse. They're often accompanied by co-occurring conditions, and around 70% of people with autism have at least one co-occurring mental health condition. So it's hard to know what's driving the results of any study reporting a difference between a likely heterogeneous group of people with and without autism. And the reason for this is likely to be because the same risk factors are shared across autism and other neurodevelopmental conditions like schizophrenia or major mood disorders, ADHD, etc. So here you can see overlapping networks of genes which increase the likelihood of a range of neurodevelopmental psychiatric conditions. And similar environmental factors such as prenatal exposure to maternal inflammation, preterm birth, prenatal vitamin D deficiency, prenatal stress, etc., all increase the likelihood of a range of neurodevelopmental conditions. And this means there's unlikely to be a simple pathway between a causal risk factor and any specific outcome. And so I suggest that we need to change our perspective, and when we think about mechanisms underlying autism or any neurodevelopmental psychiatric condition, we should instead perhaps be thinking about vulnerability and also protection. But it's actually really hard to identify vulnerability mechanisms when we carry out research in people already diagnosed with a condition. So even if a diagnosis of autism happens quite early, and that's not that common, but say around the age of 3 years old, that's actually still over 1,000 years of growth and development of the brain since birth. So to reveal the foundations for learning and memory and behaviour in individuals with and without neurodevelopmental conditions, we really need to look much earlier in brain development. But when we think about human brain development, we are usually very interested in the most complex of behaviours, such as IQ or mental health or adaptive skills. But actually, the maturation of primary sensory and motor circuits has to happen first, and brain development is then a cascading process. So there is individual variation, but certain maturational events must occur within so-called sensitive periods of infancy before more complex cognitive or adaptive behaviour is supported later on. So the genes a person has and their environment influence the path along the cascades of development that an individual takes, and they contribute to human diversity. But despite being individually unique, we recognise each other as human, and our first question in these studies, which I'm going to tell you about today, was whether there is a common framework in the very young brain which supports subsequent development. To answer that, we collaborate with Professor David Edwards and his team in the Developing Human Connectome project, and we use MRI to look at babies' brains around birth and early infancy. Now, the participants in our study have a range of likelihoods of developing autism or related neurodevelopmental conditions. And this is an example of a study carried out by one of our PhD students, Daphne Fenchel, working with Johnny O'Makirtig. So Daphne worked on the principle that when parts of the brain are growing and connecting together, they do this in a coordinated manner and at a similar rate. That means that they're correlated. So she divided the cortex of over 200 brain scans from newborns into many small equal-sized regions. And then she took 8 different measures of the characteristics of each region. And these measures included things like their thickness and surface area, the density of neural processes, and so on. She then asked what regions were correlated or similar on these clusters of measures. And Daphne came up with 7 main groups or similarity modules. And these modules made a lot of sense because they captured the key networks that we know exist in the older brain. For example, sensory motor networks or limbic networks, which looked after emotion. So this suggests that the brain's structural framework is already very well organised at birth. And next, Daphne asked if there are relationships between the different modules. And she found that yes, the older a baby is, the more interrelated or integrated their modules are. So for example, the sensory modules become more integrated with the limbic modules responsible for emotion processing. So we think we are starting to see the cascades I spoke about. Primary modules develop and start to link up with modules that look after more complex behaviour as the baby gets older. And drilling down to the level of the individual, we now have evidence that the extent to which an individual's brain structural measures at birth deviate from what's expected predicts that infant's later outcomes. And to hear more about this, I'm going to direct you to the talk by our postdoc, Rally Dimitrova, at this festival, just a little bit later on. So because individual brain development at birth predicts individual outcomes, we wanted to think more about the origins of individual uniqueness. So another of our postdocs, Judith Ziarista, has been working with Dr. Daphnis Batali, and they've been thinking about self-similarity and when it arises. So this is the idea that if I scan your brain repeatedly, there should be a fingerprint of you. That is, your brain networks at time 1 should be fairly similar to your brain networks at time 2, especially if time 1 and 2 are close together. And your brain print should be unique to you. So Judith wanted to test the hypothesis that an individual's brain print is being established and stabilised from early in development. So this is busy, but let me walk you through it. The top left panel squares show the correlation analysis between each person's brain structure and function at time 1 and time 2. And below on the y-axis, the closer to time 1, the closer to 1 the correlation value between time 1 and time 2 is, the more similar the brain measure is in that individual. So on the right, each line is an individual child. The blue dots show that the most children have a high structural brain similarity approaching 1. But their functional similarity measures in red are lower. So that means the brain structural fingerprint changes very little from 32 weeks of life, whereas the brain functional fingerprint is changing quite a bit. So why is functional connectivity so variable? Well, let's have a think about what's happening in the brain around birth. Animal studies tell us that just before birth, the cells in the cortex start firing randomly. This activity increases after the rodent is born, and cells which fire together or synchronize are thought then to be wiring or connecting together. Glutamate initially drives the increase in activity around birth, but then GABA joins in and is actually excitatory in early life. GABA later switches to become an inhibitory neurotransmitter for the remainder of postnatal life. Now, this is all very relevant to conditions like autism spectrum because gene alterations in glutamate GABA pathways or excitatory inhibitory pathways are very frequently linked to autism. So, for example, higher synchronous activity is observed in the sensory cortices of animals that carry alterations in these glutamate GABA pathways. For example, the synaptic gene neuroligin 3 is needed for these synapses, and that's shown on the left, and you can see higher activity in the animal with a knockout of neuroligin 3. And a similar pattern is seen in animals with environmental exposures linked to autism, in this case prenatal inflammation, which is known to alter the expression of GABA genes. So the pattern of functional activity in the developing brain can be altered by alterations in glutamate and GABA pathways or excitatory inhibitory systems associated with autism. Now, Judith realised that she could look at the activity in the developing human brain in a similar way using functional MRI. And here are her results of a study of functional activity in neonates. She found that in babies with a family history of autism and therefore with a higher likelihood of themselves being diagnosed with an autism spectrum condition, their functional connectivity in primary sensory regions was higher than children without a family history of autism. But have we got more direct evidence that there are differences in these excitatory inhibitory systems in autism? One way to address this is to find out if there are actually excitatory inhibitory differences in older age groups with a diagnosis of autism. And if so, might this provide an opportunity to usefully contribute interventions to individuals with autism who want or need them? And this has been looked at, but as I mentioned at the beginning, case control comparisons of people with and without autism have not been that illuminating. Magnetic resonance studies, which measure levels of glutamate and GABA in the brain, have found pretty inconsistent results when they've compared people with and without autism. And the same is also true when we scale up to look at the activity of macro-level brain networks. And even very well-powered studies find minimal functional activity differences between people with and without autism. So what's going on? Well, again, I think these sort of null effects are actually informative. And again, they tell us we should think a little differently. In particular, we should be explicitly addressing the fact that the brain is dynamic. So across multiple levels, the brain is dynamic. The glutamate GABA system that we're so interested in is in constant flux, and glutamate is a precursor for GABA. In the developing mouse visual system, we've learned that synaptic receptors rapidly adapt to neuronal activity, and the autism-associated synaptic protein, SHANK3, is needed for this responsivity. So we have asked if there are dynamic excitatory inhibitory response differences in people with autism compared to neurotypicals. And to answer that question, we've been using drugs to perturb excitatory inhibitory dynamics and asked what's the difference in people with and without autism. We first selected a drug called riluzole, which has a broad range of actions across glutamate and GABA pathways. And our PhD student at the time, Laura Ajram, found that the response to a single dose of riluzole in autism was quite different from typically developing controls. So here you can see that in response to riluzole, people with autism had an increase in their brain measure of GABA, while controls, if anything, had a decrease. And Laura also looked at functional connectivity, and she asked if activity in the prefrontal cortex is synchronized with other parts of the brain. She found that at baseline or placebo in the control group, and that's shown in blue on the left of the bar chart, there was a strong relationship between functional activity in prefrontal cortex and posterior cortices. This wasn't present in the autism group. You can barely see their blue bar at the placebo condition. However, after a single dose of riluzole, functional connectivity in autism, the green bar on the right of the bar chart, looked very much like controls at baseline. So riluzole removed functional connectivity differences between people with and without autism. Riluzole was a useful starting point, but it has multiple actions, and we wanted to begin to get to the bottom of what might be going on in excitatory inhibitory pathways in autism. So we decided to take a closer look at the GABA pathway. And I'm going to share some progress in a study of arbaclofen, which is a GABA-B receptor agonist, and this design is shown. So like the riluzole study, this used a single dose of either arbaclofen or placebo on each visit. And a core group of researchers have been analysing this data in lockdown. And I'm going to update you on the results of a visual sensory measure. So I'll leave you with the summary. As I hope you've gathered, brain structure is stabilising around birth, but brain function is quite variable. We've found that neonates who have a higher likelihood of going on to receive a diagnosis of autism spectrum have higher functional activity in the sensory cortices at birth. And this may be something to do with the formation of excitatory inhibitory circuits. And indeed, in individuals with a diagnosis of autism in adulthood, the responsivity of those circuits is altered. In particular, GABA control of sensory function appears to be altered in autism, and this is informing the mechanistically useful clinical trials which are now underway. And I'd like to offer my thanks to our funders, but most especially to all our participants. Thank you very much indeed. And I'm going to try to get back and unshare my slide screen.
Fantastic, thank you, Grainne. We don't seem to have a question yet, but I encourage people to use the chat and ask questions to Grainne, and she will stay around for a few minutes more to answer those questions. We also received a few questions or inquiries about the recording, so we are recording this event, and we will make this recording available later on. So now we can move on to the next speaker, who is Joyce Peters, is a faculty marketing manager, and she will talk about diversity and inclusion. Over to you.
Thanks. Am I sharing my slides?
If you'd like to go full screen, please, Joyce.
Okay, great. Well, thank you for inviting me to this session. It's a real pleasure to be invited to speak amongst all the kind of people on this panel, so thank you so much. My name's Joyce, and I am the Faculty Marketing Manager for the IoPPN, but I'm here to talk to you today in my capacity as the Race Equality Co-Champion, and I share that role with my colleague Hui. We were appointed towards the end of last year, and I also stepped down recently from the Race Equality Network chair, so you may have seen me in the Race Equality Network meetings that we run every month. We have a great D&I team here that do a lot of fantastic work. Zoe Kennedy, Professor Stephanie Hatch and Professor Anne McNeil work across the 9 protected areas, and they work alongside champions and other volunteers to do that work, and that's how I'm involved. And I would really recommend taking a look at their pages and reaching out to Zoe if you have any interest in finding out more about the full breadth of their work. So I'm going to talk about why diversity and inclusion is a topic for everyone. So in May 2020, McKinsey released their 3rd D&I report, Diversity Wins, which looks at the business case for diversity and inclusion. The latest report shows not only that the business case remains robust, but also that the relationship between diversity on executive teams and the likelihood of financial outperformance has strengthened over time. The report found that diversity and inclusion can enhance the problem solving necessary to rethink businesses and reimagine industries in the face of unprecedented disruption. And more diverse teams are better at anticipating changes in consumer needs and buying patterns, which can lead to more rapid product service innovation. So this report and others show us that our underpinning assumptions about life determine everything we do, including our work and how we relate to others. So it's really important to challenge ourselves and test our assumptions, especially in the kind of work we do here at the IoPPN. In June of last year, our executive dean, Professor Ian Everall, set the expectation that every member of the IoPPN actively engages in work to counter racial inequalities within their teaching, research and interactions across the IoPPN. The principles of equality, diversity and inclusion are at the heart of the IoPPN's culture, and as an organisation we are committed to continuing to discuss, implement, promote and monitor strategies aimed at improving the working environment for all staff and students. So with all that being said, what can you do? So if you don't know how or where to start, that's okay, because we have tools and resources to help you get started. And it is a journey. Once you start, you know, there's no end point. You're just going to keep learning and growing in your understanding as you continue. So we have the community engagement corner, and that is a really good place to start. It's got loads of really good resources, training, things to read, and it's a good jumping off point where you can go and find more information. As mentioned, the Race Equality Network, that's a great place to go. We have monthly meetings, and in those meetings you can ask questions. There's time for discussion. Staff and students are welcome. People often come with ideas or things they want to try and do, and they kind of can get feedback from their colleagues and learn from each other. The Race Equality Network is also responsible for the Black History Month event. So if you've seen those around, all of that work is done through the network, and so we kind of try to do learning through culture and celebrating culture, and the network is always looking for new people to join, and we can only celebrate as much as we know about. So getting involved and learning through culture is really important, and we can only do as much as we know. So the more diverse the group, the better for the work we can continue. And something new that we're launching this spring, the Year of Learning. So the Year of Learning is an initiative for everyone to embark on their own journey of self-reflection and learning around race and ethnicity, and how this intersects with other protected characteristics. Each month, participants can do, read, watch, find, experience or research something that helps them grow and explore their own relationship to race and ethnicity in the UK and around the world. Learnings will be shared via the dedicated Year of Learning community engagement corner page and through other CD&I channels. Together, participating in this journey will demonstrate how small moments of learning and reflection can together support large, impactful change in addressing current racial inequalities and ensure IoPPN and Kings are inclusive environments that enable everyone to thrive. So I'm trying to be quick, but I want to leave you with a question, and that question is, what is racism? What is that experience? Is it something bad? Is it uncomfortable to talk about? Maybe hard to quantify? An excuse that people use? Personal opinions or structural issues? Widespread or hardly there? This isn't a question that I'm going to answer right now, but something I'd like you to go away and think about. I could provide a definition, but true understanding really comes from trying to find that answer for ourselves and really putting ourselves in that position. Diversity, inclusion and equality are woven into the culture of an organisation, and everyone in that organisation creates that culture, which is why D&I is something we should all be invested in. D&I work isn't a nice to have. It lays the groundwork for better decision making, higher engagement, greater impact in our community. And if we pay some attention to these areas, we can produce better work. And ultimately, that's what we all want to do here. So thank you for your time. I can answer any questions that are in the chat, and I can post links to the resources that I've mentioned. Thank you.
Many thanks, Joyce, for highlighting the diversity, inclusion, and equality work that is happening here across the IoPPN, and also why it matters. Our next speaker is Dr. McAdams is a Wellcome Trust Senior Research Fellow. Over to you, Tom.
Okay, thank you. So hi, I'm Tom McAdams. I am a Senior Research Fellow based in the Social, Genetic and Developmental Psychiatry Centre. Okay, so we all know that mental health problems run in families, but what we don't know is why. So there's at least 4 reasons why this might be happening. So first, exposure to mental health problems in parents may cause the development of problems in children. Second, the transmission of genetic risk from parent to child may be explaining associations between parent and child mental health problems. Third, shared environmental effects that influence the mental health of both parents and children may explain why they both experience these problems. And fourth, it's possible that child mental health problems cause mental health problems in parents. Okay, so these 4 possibilities they're difficult to distinguish between, and this is basically what my research revolves around, trying to understand why mental health problems run in families. In order to do this, we need to use quasi-experimental research methods. So these involve things like adoption studies, where children are not related to their parents, sibling comparison studies, in which siblings, they share a lot of risk factors, they share a lot of confounding variables. So when differences in exposure between siblings predict differences in outcome, then that gives us reason to draw causal inference about associations between exposure and outcome. And another method we use is extended families. So these are families comprised of twins or siblings with children of their own, and we can use these families to disaggregate parent and child associations into parts attributable to causal effects versus genetic effects. Okay, so for the last few years, I've been working on the MOBA dataset. So this is a huge Norwegian database, 90,000 families recruited during pregnancy, followed across the life course. Children are currently in their teens, and we have lots of information on the mental health of MOBA participants. It's an amazing data set to kind of ask these sorts of questions. What we've done is we've linked MOBA to the birth records, the birth registries that we have in Norway. And in so doing, we've identified tens of thousands of extended families within the MOBA database. We've then developed statistical models to apply to these data in order to ask questions about why do mental health problems run in families, why are particular parental traits predictive of mental health problems in children. We've applied these models to all sorts of different questions. I just want to give you a couple of examples today. So it has been suggested that exposure to maternal depression during pregnancy somehow causally programs the developing fetus in such a way that leads to the development of emotional and behavioural problems in those children as they get older. What we've shown in MOBA is this isn't the case. These associations are actually attributable to genetic relatedness between parent and child. There is no causal effect that remains. Postnatal exposure to maternal depression, however, does persist after we account for genetic transmission, and this effect appears to get larger with age. So a really interesting, exciting thing about MOBA is that they've actually collected DNA on all of these families as well. And this is something that's happening more and more within science, is that we're getting these family pedigree-based studies that are collecting DNA. We're also getting biobanks that collect DNA that happen to have big families within them. And we're getting this merging of methods. It's a really exciting kind of time. It's going to, I think, teach us all sorts about intergenerational transmission, about the etiology of mental health problems. So I'm writing a review on this at the moment. This will be out later in the year in JCPP, but I think this is a research area to watch for sure. So finally, I just wanted to highlight, so earlier this year I started my Wellcome Trust Senior Research Fellowship. This is 5 years of funding to explore all sorts of things that I'm interested in. So I'm very excited about my plans. So I just wanted to share some of the plans that I'll be kind of getting stuck into over the next few years. So first is treatment effects. So we know that exposure to mental health problems in parents is detrimental to child development. What we don't really know very much about is whether treatment of mental health problems in parents actually has positive consequences for children. Does it mitigate this transmission or not? I think this is a really interesting and kind of important research topic, and it'll be interesting to learn more. I'm also interested in genetic sensitivity. So we know that some children are more susceptible than others to environmental stresses. Some are more resilient than others. What we're doing is we're going to try to identify genetic variants associated with this sensitivity. So our end goal is to create polygenic scores, basically that index sensitivity to the environment. This is a really interesting research area. Third is selection bias. So we know that people who volunteer to take part in scientific studies are basically different to those who don't volunteer to take part in scientific studies. And this skews our understanding of the things that we're interested in. We're only studying them in volunteers. So this biases everything that we're learning. So I'm really interested in trying to understand this bias and explore ways in which we might be able to correct for it. In the MOBA, we can link to kind of birth records, to medical records on absolutely everybody, whether or not they took part in the study. So this gives us a window in, a kind of route in to try to explore the kinds of biases there are and how we might kind of correct for them. Last but not least, I just wanted to flag the Children of Twins Early Development Study. So this is a study that I've been running for the last 5 or 6 years. This is basically, we're collecting data on the children of those twins who've been taking part in the TEDS twin study for the last 25 years. So the aim here is to create a second generation. So we have 2 consecutive generations followed from birth into development, and we can use these databases to explore, why and how intergenerational risk is kind of transmitted from parent to child, and yeah, this is the Children of TED study. So thank you very much for listening.
Thank you, Tom. Thank you for keeping also on your presentation within the time limit. We are actually running a bit ahead of time, which is unusual for academics to be even ahead of time. But so we are, I'm going to introduce the next speaker, who is Professor John Marsden. He is Professor of Addiction Psychology. I can see his slides are ready, so over to you, John.
Thank you so much, Paolo, and I really appreciate the opportunity to take part in the Research Festival this year, and it's great to see so many people joining us for this amazing event. And I'm particularly pleased to shine a brief light on a problem that is very vexed for us clinically, and that's the treatment of cocaine addiction. And I'm going to summarize very briefly some of the work that we've been supported by the BRC to do locally, which we hope is a developmental stage for further trials in the future. And I'm going to focus on the topic of cocaine addiction in the context of mental imagery and how we can help people better control craving. Cocaine, it's a very powerful addictive stimulant. There's over 18 million people using this drug across the globe each year. In this country, in England, we have 1 million users of powder cocaine each year, and 1/3 or so of people using a very addictive variant of cocaine called crack. It's an alkaloid smokable variant. There's a picture of powder cocaine on the left there. Each year, around 100,000 people attend NHS and other NGO specialist clinics seeking help for cocaine use disorder. And this is a real problem because we don't have any approved medications. Overdose from cocaine fatalities are increasing year on year. We have an array of psychological treatments. They are effective, but as we heard about effect sizes, they're only weakly so, and we need much stronger treatments. When people are exposed to cocaine, about 1 person in 20 becomes dependent in the first year. And then if you look over the lifetime, about 5% overall become dependent. So it's a very addictive drug. And even with the treatments we've got at the moment, 80% or so of people relapse in the first 6 months. So if I could have the next slide, please, Trevor, thank you. So as with many psychiatric disorders that are disabling and maintaining, the main puzzle is why do they occur? And as one might expect with cocaine, there is an absolutely fundamental exposure-driven adaptive learning process. So it's impossible to be addicted to cocaine through one exposure. And in fact, when you talk to patients that we see clinically, as you might imagine, they acquire over time a progressive involvement in drug taking that becomes very central to their lives. And so hundreds and hundreds and hundreds of hours are devoted to obtaining, consuming, and often recovering from the drug's effects. But cause and maintenance is tricky. We take a very individual view, but it does appear that very quick forming memories, episodic memories of cocaine reward are laid down early on when people are exposed to this drug. Lots of neutral things, for example, the ATM machine I photographed locally on the left there, completely drug neutral to us, but become very powerfully associated with cocaine use for a person that has cocaine use disorders, and have the capacity when encountered of triggering very powerful, often sensory, visual and other sensory mental processes that signal drug wanting and expectancies about using the drug. And those fast arriving mental images of drug taking really take a person unaware. They in a way, they occupy the contents of consciousness so that the rest of the world almost melts away. And the person often finds it very, very difficult to shift their focus away from that mental image. And unfortunately, the mental image acts as a hook. It brings to mind other episodic memories from the person's autobiographical memory filing cabinet, if you wish, previous drug use experiences. And then there's a future tense as well. The person starts to think about how the immediate future of their life would be well served by using the drug, and negative effects about drug use experiences are discounted in favor of positive effects. And that can sometimes take place in a very implicit or almost automatic cognitive process. So the person is almost surprised that they've suddenly initiated the sort of complex behavior to buy the drug, mainly involving speed dialing a drug seller. And other times it's a very distressing and protracted process where the person is desperately trying to not yield to the urge to obtain the drug, and may or may not be successful, usually not successful. So how do we help people better deal with these impulses and desires? And fundamentally, how can we help people control craving? So I just want to, before I go to my last slide, just to say that we have been taking baby steps here, working very successfully with a small number of patients, and we're so grateful to our local patients for collaborating with us. And in psychiatry, if you think, no matter what the problem is, there is the word avoid on one end of the spectrum and approach on the other. And we've been grateful that our patients have been very keen to approach very distressing thoughts, to expose themselves to triggers and reminders of drug use, in order to see if we can help process and better control craving. My last slide, please, Trevor. Thank you. So one of those cheeky opportunities to put 3 slides in one, but I'll be very brief. We've had the audacity to develop a novel cognitive therapy that adapts an evidence-based approach to help people with post-traumatic stress disorder, which, if we could possibly just go back to my, thank you so much, I won't be too much longer. And PTSD is disabling fear triggered by sensory matched cues to a experience of either a life-threatening experience or the witnessing of one. And there are many similarities with addiction, which I won't belabor now, but the activation of a consolidated craving memory renders it labile to alternate appraisals, updating using cognitive restructuring, and a variety of other toolkit techniques that we've used, and I've summarized in that slide in the middle, where a patient is able to think forward in time as to the outcome of a drug use episode and use that negative knowledge as a way of countering a live craving experience. It's baby steps. So we randomized just 30 patients to an assessment only, an exposure only condition, or a condition in which those exposures additionally included an intensive program of cognitive therapy. And our patients were very happy to take part. We gathered some encouraging clinical evidence. We've put this memory-focused cognitive therapy into and among other therapeutic tools that we use. And we're now hoping to take this forward and combine this psychotherapy approach with what look like a couple of very exciting candidate medications for cocaine use disorder. And in the addictions field, really combination therapies, bringing in and melding together medications and psychotherapy looks like a very exciting territory, and one that we hope to pursue in the coming years. Thank you so much, everyone.
Thank you, John. Thank you for giving this comprehensive picture on cocaine addiction. So we are back on time at the moment, so we move swiftly to our next speaker, who is Tatiana Salisbury. She's Senior Lecturer in Global Mental Health. Tatiana, if you are ready, you can start.
Apologies. Hello, everyone. Okay, so I'm here to talk about how we might improve mental health and what we do when we find out that expertise actually isn't enough. So cultural and contextual factors that are important when thinking about mental health. And they're important because they help us to determine whether or not symptoms are present, as well as whether or not a diagnosis of a condition can be made. So this alone gives us reason enough to include those who would either receive a treatment or service, as well as those responsible for actually delivering those services and treatments in discussions and in planning for the development, the implementation, and the evaluation of interventions. However, traditionally, these groups have been excluded from meaningful participation in these activities. And this is not a surprise, as the development of interventions focuses really heavily on questions around theoretical grounding, around fidelity and feasibility. Within the Medical Research Council's framework for the development and evaluation of complex interventions, researchers are asked to consider what types of user involvement they will include in their work. And these aren't explicit questions about whether or not there'll be a demand or uptake of the interventions by those who would either receive it or be tasked with delivering it. And so participation then is often confined to activities around dissemination and recruitment, as well as rubber stamping interventions, which have largely been finalized, rather than having stakeholders engaging in respected conversation and voice and choice in its design, content, and evaluation. So there's a large gap in our understanding about how to meaningfully partner with users, with service providers and communities and also about what the impact of actually partnering with and engaging stakeholders has on the resulting interventions and their effectiveness, their sustainability, and their scale. So my work focuses on improving the mental health of teenage girls during pregnancy and in the year after birth. And these girls are living in Mozambique and in Kenya. And this is through the UKRI Future Leaders Fellowship funded project Inspire. And how we aim to address this imbalance is by actively engaging with young women, with their families, with health and community providers, and also with government representatives in order to understand their needs and understand what it is that they see as important to them and their communities. And we do this through a blending of different approaches: human-centered design, systems thinking, and implementation science, with an aim to deliver an intervention that is designed for impact, sustainability, and scale. So Inspire is a collaboration with the International Center for Reproductive Health in Mozambique, with the Aga Khan University in Kenya, the Helen Hamlin Center for Design at the Royal College of Art, and the Center for Global Mental Health here at King's College. And what we are doing is we're working with local stakeholders in order to identify and deliver a workable prototype. And once we've got that prototype up and ready, we will then test it out on small numbers of young women to understand exactly how it works and how we can make it better. And after a few iterations of this process, we hope to then have an intervention that is ready for testing through a trial, in which we can then examine the effectiveness it has for improving mental health outcomes, as well as assess its impact on other adolescent outcomes and on the outcomes for the communities in which these girls live. So in closing, I want to say that complex interventions do not necessarily require a complex solution, but they do require the input and the collaboration of people with different perspectives and different experiences. Thank you very much.
Thank you, Tatiana. Thank you very much for keeping your presentation within the 5 minutes. Our next speaker is Dr. Matthew Grubb. He's reader in Neuroscience. Let me check if do we have the slides for Matthew? There you go. Okay, I can see the slides, so I can move on.
Hi, I'm Matt Grubb at the Centre for Developmental Neurobiology, where we're getting nosy about brain repair. Now, brain repair requires more than regrowth. Here we've got a very generic network where input comes into a particular target circuit, which then generates some kind of output. And if for whatever reason this input degenerates, then of course the first thing that needs to happen to repair all of this is that that input needs to regrow back in. But that's not enough. The input then needs to reconnect to the target circuitry, and then the target circuitry needs to reuse that input in a useful way to generate a useful output. Now, the problem in learning these second and third processes is that there aren't many bits of the brain that can do the regrowth in the first place. Getting axons to regrow is very difficult. So what we need is a system that can regrow and repair itself naturally so that we can start to understand these later processes. And we studied the olfactory system for that reason, and that's why we're getting nosy. We studied the first projection from the nose up into the brain, where there are neurons called olfactory sensory neurons, which detect information in the outside world and send it up to the brain. And these neurons, for lots of reasons, can end up being damaged, but as long as there are stem cells left in the nose, those stem cells can repair that whole population of neurons, and they can regrow back up and re-establish themselves into the brain. That all happens naturally, so completely natural regrowth. And our question is, what happens to the reconnection and the reuse of this information? And by studying this naturally occurring brain repair, we want to learn lessons for potential clinical interventions when we're trying to induce regrowth in other parts of the nervous system. And to study this natural reconnection of inputs into target circuits, what we use is a very simple toxin-induced degeneration model, where to start with we can follow the anatomical connections of these projection neurons into the brain, and this is a visualization with a particular marker called OMP of those projections. And what we see is that after we give the toxin which kills off these cells, then they degenerate over about 10 days, but then we see them come back. And there's a really sharp reconnection phase of about 2 to 4 weeks after the degeneration starts, where these connections start to reform. And we're starting to look at their functional properties while they're reforming. What are the electrical properties of the connection between these regrowing fibers and the cells they target in the brain? And we use a slice preparation to look at this, and we record electrical responses in the cells in the brain. And we look at patterns of those responses to tell us something about the properties of the inputs as they come in. If we give a pair of stimuli to the incoming inputs and we record a pair of responses to those stimuli, under normal circumstances, the second response is much smaller than the first one, and that actually indicates that this connection is pretty strong. But what we see when the inputs are regenerating or reconnecting into the circuit is that that second response is about the same size as the first one, and actually what that tells us is that these reconnecting nerve terminals are not working as strongly as they would under normal circumstances. So we have weaker neurotransmitter release in these reconnecting fibers. So then we want to know what the target circuitry is doing to reuse this incoming information. And we're studying this simple circuit where the input from the nose is coming into the brain and being used and being sent out to higher areas such as olfactory cortex. And we know from what I just showed you that the inputs are weaker during the reconnection phase. But does that mean that the outputs are weaker too? Well, what we can do is start to look at the local cells which are involved in processing this information. And there are local neurons which boost the strength of the signal from nose going out to cortex. And we know that actually their activity is stronger during the reconnection phase. And there's another set of cells which are inhibitory, so they put a brake on the strength of the information coming from the nose through to cortex. And those guys are actually more weakly active during the reconnection phase. So how does that all add up? Well, if we look at the overall relationship between the input and the output of this circuit, then what we see is that that's actually stronger. So we get more activity out for a given amount of input in. And what we think is happening is that there's compensatory plasticity in the network, which enables the target circuit to use the information coming in even though it's weaker. And we think that might be really important to try and look at potentially inducing the same kind of plasticity in places where we're trying to get inputs to regrow in other areas of the nervous system. And who knows, that might help brain repair in the future. Thank you.
Thank you, Matthew, for your presentation and showing us the work you're doing. So our next speaker is Dr. Alika Dimitrova. She's a research associate, and she will be talking about neonatal brains. Alika, are you ready?
Yes, sorry, I'm just, yes. Can you see my screen?
We can see it, yes.
Perfect.
You should be, yeah.
Yeah, great. So virtual welcome, hello from me. Sorry? Oh, okay. So first, just a big thank you for inviting me to give a brief summary of our recent work of why we should talk more about heterogeneity in early brain development. Fetal and neonatal brain development represent the most dynamic stage of human brain development across the lifespan, during which period there's some very dynamic cellular events that establish the fundamental structural and functional organization of the human brain and promote the emergence of cognitive functioning. During the very narrow time window between conception and birth, we see some substantial changes in the volume, the shape, and the microstructure organization of the brain. And given this complexity, it's not really surprising that even small deviations to one or multiple of these processes could actually result in atypical neurodevelopment. Preterm birth is a well-recognized risk factor that has been linked to abnormal brain development, as well as a wide range of cognitive and behavioral difficulties in later life. And even if there have been some significant advances in our understanding of how prematurity really shapes the developing brain, the predictive power of neonatal MRI remains relatively poor. So we can't really say if a preterm infant will develop typically or whether they'll have some difficulties in later life. And this in part could be due to the fact that the preterm population is described by a very high clinical and biological heterogeneity. And the most widely used approach to study brain development, which is the group-wise or group mean comparison studies, by definition really focus to describe the group average, and in that way it kind of fails to capture individual differences that might be significant to a given individual and their later outcome. And therefore, having an approach that can really help us model or capture this heterogeneity could better our ability to predict outcome in later life, especially in infants which are at high risk of atypical development. And a widely used approach in pediatric, for example, is to compare the individual to a robust normative populational data set. Using growth charts of baby weight, height, or head circumference, we can actually assess the development of individual infants against this normative mean and the variation associated with it, taking into account the baby's age as well as the baby's sex. And we can quantify this as a deviation above or below this populational mean. And of course, this can easily be translated to neuroimaging. We can use a large, healthy term-born sample of infants to model the developmental trajectory of any other given imaging feature using age, sex, or any other predictors of interest. Once we have established what the typical trajectory should look like, we can then offer a personalized assessment of how the brain develops in individual infants. And we can, of course, quantify it in units of standard deviations or Z-scores above or below the mean, given the baby's age and sex. And just a few very brief examples here. So brain microstructural development is often being reported atypical following preterm birth. Using this framework or normative modeling, we can actually characterize what the typical brain should look like for a given age and sex. Then we can use this to localize areas of the brain where we can see abnormal development at an individual level. So here we have 5 preterm infants, all scanned at the same time and the same sex. And highlighted in reds are areas where we see abnormal development, which is more than 3 standard deviations away from the mean. And hopefully it's visible that this atypical development is highly heterogeneous in terms of the spatial extent as well as the location, and that while some preterm infants show no impairment, others actually show well-spread atypicalities. And this variability across individuals, again, the same age and the same sex, could possibly explain why the findings of group-wise studies have been so inconsistent. And of course, the approach could be used in any other imaging features, so we can map the normative trajectory of the development of ventricular volume, for example, to then identify infants with extreme ventricular dilation, more than 10 standard deviations away from the mean that's expected for the age and sex. And it so happened that actually those 5 infants have very poor neurodevelopment in later life. So to use normative modeling to offer an individualized assessment of brain development could be a very powerful tool. And this could vary from some more basic, but of course not simple, clinical applications of automatic detection of areas of the brain where we see very abnormal appearance. And those incidental findings are actually very frequently seen in preterm infants and often have long-term lasting effects on neurodevelopment. And of course, we can actually use those normative charts to monitor the progression of brain development in individual babies, and especially in those infants who we consider at high risk of atypical neurodevelopment, and whether that's because they're born preterm or because they have familiar or genetic predisposition for it. And lastly, of course, by kind of moving away from the mean and the average patient and focusing on the individual and their very unique clinical circumstances, this personalized approach could better the use of neonatal MRI to predict later function outcome and specifically identify infants who might require further clinical or behavioral therapies. Thank you.
Thank you, Ralika. Thank you very much for your presentation. So our next speaker is Kuri Kim. She's a PhD student funded by the MRC Doctoral Training Partnership. She's one of the 3 Minos thesis finalists here, the IoPPN, and one of the hit winners. She has also recently won the King's College London final and will go on to represent King's at the national finals. And I believe this is the first time that we have a PhD student from IoPPN at the national event. So congratulations for that achievement. So over to you.
Do you ever drive to the supermarket to pick up a few groceries only to come back out and completely forget where you parked? How about when you want to leave your house? Where did you put your keys? Surely you wouldn't have put them too far from where you normally do, but why are you struggling to find them right now? And if you can relate, do you find that this is happening more and more with age? This decline in your memory is a very normal part of getting older. But what if I told you that there may be a way that you can slow it down or prevent it from happening altogether with a change in your lifestyle? This is what my PhD is trying to do. For the longest time, it was thought that brain cells were unable to regenerate. Once they died, there was no coming back. However, we now know that there is actually a small part of your brain called the hippocampus, as shown by the red in the image, that is able to continue generating new brain cells throughout life. These new brain cells will then integrate themselves into your brain, and this is a process called adult hippocampal neurogenesis. Neurogenesis is thought to play a role in memory, in particular a type of memory which allows you to tell the difference between 2 very similar events, for example, where you put your keys every night. And this memory will start to get worse from around our mid-20s, with a sharp decline from around 60 years old. And not only that, but as we get older, neurogenesis also slows down and we produce less brain cells. And we think that this might be part of the reason why our memory gets worse with age. Our lifestyles and diets have a huge effect on the way our brains function. And intermittent fasting, a diet where you go through periods of fasting and normal eating, may have a positive impact, particularly on neurogenesis. And I wanted to investigate this. So for my PhD, I asked a group of older adults aged 60 years and above to go on the popular 5-2 diet. This involved normal eating for 5 days and 2 days of only eating food packs I provided them, totaling 600 calories a day for 12 weeks. And for this trial, I tested their memory before, during, and after the diet. And the results we got are very encouraging. Those who are on the diet saw an improvement in their memory test, as shown in this graph. And this was not seen in those who were just asked to continue with their normal routine. This means that intermittent fasting could really improve your memory, possibly by boosting neurogenesis. By 2030, 1 in 6 people are expected to be aged 60 years and above, and this is likely to have a huge impact on our society. But if there are lifestyle changes that can be made to prevent or delay this decline in your memory with age, or even the onset of age-associated brain diseases such as Alzheimer's disease, this could have a huge impact on our healthcare systems, the economy, and most importantly, yours and your loved ones' quality of life. Thank you for listening.
Thank you, Kuri. I can see why you've been the winner of the heat and the King's College finals. I will definitely look into fasting if it will help my memory. So we can now move on to the next speaker, Dr. Catherine Young. She's the NIHR Moseley BRC lecturer. So over to you, Catherine.
Great, thanks so much for having me today. So yeah, I'm a BRC lecturer based at the SGDP. And I'm going to be telling you a bit today about 2 mental health studies we've been running during the pandemic. The Repeated Assessment of Mental Health in Pandemics, or RAMP study, which I lead, and the COVID-19 Psychiatry and Neurological Genetics study, or COPING, led by Professor Grainne McAlonan. Our studies are conducting regular monitoring of mental health in a sample of over 30,000 individuals across the UK, aged 16 years or above. In RAMP, we recruited participants at the beginning of the pandemic via social media, whereas in COPING, we recruited participants from existing recontactable cohorts through the NIHR bioresource. And these studies include a lot of different outcome measures. We're looking at a range of mental health conditions as well as respiratory and neurological symptoms of COVID. But my main interests in this work are to examine socio-demographic and potentially modifiable risk factors that influence long-term trajectories of anxiety and depression. So when I mention modifiable risk factors, I'm interested in certain cognitions or patterns of thought that people engage with and certain types of behaviors to understand how these types of processes influence changes in anxiety and depression during the stress of the pandemic. Today, I'm going to share with you a little bit of data from a subsample of our participants who had pre-pandemic mental health measures available as well. These are individuals with pre-existing diagnoses of depression or anxiety and were members of the Genetic Links to Anxiety and Depression, or GLAD study, which is one of the recontactable cohorts within the NIHR bioresource. Within this sample, 62% reported a perceived worsening in their mental health from before to during the pandemic, when asked in around May last year. This was in response to a single item question asking, how different do you feel your symptoms of depression or anxiety are now compared to before the pandemic? However, when we look at changes in symptom measures, here using the PHQ-9 for symptoms of depression and the GAD-7 for symptoms of anxiety, we see a slightly different story. We see very little overall change in symptoms, and if anything, a slight overall decrease from before the pandemic to the early months of the pandemic last year. So what this suggests is that whilst quite a high proportion of individuals are experiencing a worsening in their mental health and some level of psychological distress, this doesn't necessarily directly map onto an overall increase in symptoms across all individuals. So this is something we're continuing to explore more, but one of the reasons for this might be in relation to how well we remember our past symptoms. If we underestimate how severe our past symptoms were, we may experience a worsening in our mental health, even though overall symptom levels maybe don't change that much. Of course, looking at overall means is only part of the picture, and within this there are many individuals who did experience a worsening in symptoms, as well as many who experienced an improvement of symptoms over this period. So we've been investigating individual difference effects to try to identify who are the individuals at greatest risk of worsening mental health over the course of the pandemic. Here we're looking at prior mental health diagnoses, and what we find is that both comorbid depression and anxiety, as well as depression only, and various other diagnoses including eating disorders, obsessive compulsive related disorders, PTSD, autism spectrum disorders, and personality disorders are all associated with a greater risk of worsening depression and anxiety from before to during the pandemic. We've been also looking at a range of demographic factors and find quite a striking effect of age, where particularly young individuals, here aged 16 to 25 years, are at elevated risk of worsening depression and anxiety from before to during the pandemic. This is in contrast to individuals aged 46 years or above, who were actually more likely to experience a reduction in symptoms over the same time period. In relation to other demographic factors, we also find an effect of female gender, of being a student or of being unemployed are also all associated with a greater risk of worsening symptoms. Moving forward, we hope to examine these changes over the longer term. We're continuing to collect data and have monthly data on our entire sample, and we'll be starting to explore the cognitive and behavioral factors that predict differential trajectories over the long-term course of the pandemic. Thank you.
Thank you very much, Catherine, for this very topical presentation and showing the impact that the pandemic is having on people, especially young people. So our next speaker is Chris Albertine. He is a research portfolio lead for the Department of Old Age Psychiatry. And I can leave the floor to Chris. I see he's ready. Over to you.
Thank you, Paolo. Hello, everyone. Given the theme of this year's festival, I just wanted to take a second to state what a lifespan perspective means to me using this quote. If we operate with a belief in long sweeps of time, we build cathedrals. If we operate from a fiscal quarter to fiscal quarter, we build ugly shopping malls. Now, this ethos is a driving force in my work, and I'd like you to bear it in mind for my talk. I really want to stress the urgency of the dementias here and why they're such formidable opponents. They're incredibly debilitating and distressing to individuals and those closest to them, and they consistently lead in mortality, and yet they have no effective treatment to slow, prevent, or cure the diseases. They're exceptionally high cost. And this last one, as a trialist, kills me because 99% of therapeutics fail to make it through the trials pipeline, and 99% of potential eligible participants in the community are not even referred to clinical trials. I mean, it sounds like I'm making it up, but these are genuine statistics, and we have to do better. So if we can collectively mobilize with purpose, as this last year has taught us, there are no more excuses. Now, a primary reason for these statistics and the challenges facing dementia research is the complexity of mechanisms underlying neurodegeneration and the heterogeneity of the patient population. However, with this growing wave of digital and data, it's providing us with opportunities to find those impactful inflection points. Now, essentially, artificial intelligence is an optimizer. It digests high-dimensional data to recognize patterns and automatically derive actionable insights by suggesting the path of least resistance. It also has a memory, so it builds on previous inputs and stores them, so very appropriate for progressive diseases. Certain models are autodidactic. It teaches itself, innovating without direction, a real in silico ally. And it's been showing promise in many areas. Including in clinical assessments in my department, in imaging, health system optimization, innovative drug discovery and biomarkers, but it's still very, very early and controversial. It's in its infancy still, and like an infant, it really needs appropriate nutrition, exposure, and guidance for it to grow into its best self. So in other words, it needs access to detailed and validated data and subject experts. This was confirmed just over a year ago when we, through the RIC, we ran some AI and mental health workshops where the overwhelming consensus were that there were issues regarding availability and visibility of richly phenotyped data and access to those clinical and subject experts. So I thought to myself, what can I do to facilitate expert networking and data access? Introducing ARC, Aging Research at King's. Now, in my 10 years in research, ARC is the best example of interdisciplinary cooperation with a common goal that I've ever come across. It's led by Dr. Richard Sial in Folsom, and ARC takes a very holistic approach, bringing together expertise across all faculties of King's to tackle age-related issues. From basic and clinical sciences to sociological impacts, mathematicians, medics, architects, programmers, entrepreneurs, you name it. It's a wonderfully eclectic ecosystem. Now, in 2019, we launched the Longevity AI Consortium, which is specifically targeting data science innovation in healthy aging, with several projects ongoing and starting up. I'd like to highlight in particular ARC's focus on knowledge exchange to the community through key stakeholder partnerships. For example, their policy links, membership on all-party parliamentary groups, advising public health initiatives, and linking with the Parliament's knowledge exchange unit. But also commercially, it's partnering with internationally with companies in tech, pharma, and the consumer space to facilitate sustainable innovation in society. Secondly, alongside a fellow speaker today, Dr. Petra Protsi, we are the joint Demon Network King's regional leads. Now, Demon comprises of an international network of over 1,000 members, including clinicians, researchers, and data scientists, all innovating to combat dementia with data-driven insights. It's supported by DP UK, which provides access to over 3 million richly phenotyped individuals from 42 cohorts, and the Turing Institute, providing bespoke data science expertise. It's structured by having focused working groups targeting key areas, such as the biomarker working group led by Petra, or trial optimization, drug discovery, prevention, and more, all collaborating on analyses, papers, grants, data sharing, and expert reviews. ARC and Demon have partnered together with this common goal, and it's open to all, so please do check it out. And finally, the IoPPN really does, as we just heard, have a wealth of data collected over the years. It's just hard to find sometimes. So with the help of some of my RIC colleagues, we asked around for some old and current cohorts relevant to mental health and neuroscience, and created a directory. The handy thing for an aging and dementia researcher such as me is that the main variable you require is age, and that is ubiquitous across the cohorts. I'd like to just particularly credit Professor Craig Morgan and his team for helping me jumpstart this, because really couldn't have done it without him. And please do get in touch if you want access. We owe it to the professors of our past to maximize the value of this data and deliver for patients and the public. So in summary, if you'll permit me just to return to the original lifespan perspective analogy, through ARC, Demon, and RIC, we are bringing together a critical mass of architects, workers, raw materials, and an ever-evolving sophisticated tools. So much like the construction on campus right now, we are laying a firm foundation for innovative data science and machine learning methods with optimal patient outcomes at its core. But we always need more. So this is an invitation to all who want to be part of the biggest medical research challenge of the century. Come build cathedrals with us. Thank you.
Thank you, Chris. Thank you for highlighting the work you're doing, and the resources and the research on aging and dementia that is happening here at the IoPPN. So our next speaker is the last speaker, but not the least, for our first session of this afternoon festival. She is a senior clinical lecturer and Dr. Marina Dutta.
Thank you. Yes, I'm going to be talking today about data science and how it can be used to understand suicidality and self-harm in the digital age. Now, suicide is a global public health concern across the lifespan, with close to 800,000 people dying by suicide every year worldwide. And by far the strongest risk factor for suicide is a previous suicide attempt. We also know there's been a worrying increase in self-harm in recent years, particularly steep amongst teenage girls. So ever since I've been at the IoPPN, I've studied suicide and self-harm behaviour, initially using epidemiology research methods, but latterly I realised that to advance the field of suicidology in psychiatry and study dynamic events in each patient's life course, what I needed was to use data-driven computational methods such as natural language processing and machine learning within the precision medicine paradigm in order to try to personalise monitoring and care of patients. So why is this? Well, what are the challenges with understanding suicidality? Most research into suicide amongst mental health patients has been based on static risk factors. These are represented on the left of my slide. These have been studied in case control studies. So, for example, life events such as bereavement, being bullied or ostracised, simply being of the male gender, having a family history of suicide, a comorbid mental health problem, or being on, for example, opioid painkillers and having a cancer diagnosis. All these things are known to affect someone's risk. But predicting suicide is very much like looking for a needle in a haystack, and that's because of the low population base rate of suicide, but also the low positive predictive value of any prediction tools or scales to date. And although these static factors give us an indication of an individual's general propensity to be at risk for suicide, they really don't capture the fluctuating nature of risk. And for this, shown by the schema here of a lifetime perspective, we really need much more subtle, time-dependent dynamic risk factors to be studied. And these factors, symptoms, behaviours and mental health service use, have really been hidden away in the free text sections of electronic health records. And I really wanted to study this. So over the last few years, my work has gone into this study funded by a fellowship from the Academy of Medical Sciences and Health Foundation called Electronic Health Records to predict hospitalised suicide attempts targeting information technology solutions. And it's heavily relied on the fact that clinicians have been inputting data to electronic health records in our trust since about 2006. These are then anonymised and available through the case record interactive search. And we have been so fortunate to create an infrastructure with a lot of hard work from many people to data link with hospital episode statistics, providing information on admissions to hospital, but also with data from the Office for National Statistics on mortality. And the aim has been really to study this data in close detail, particularly the free text. Ultimately, it would be excellent, and what I'm looking at next is to begin to feed back to clinicians and provide some sort of decision support. But I just wanted to give you a flavour of the sort of work that is very labour intensive. It's multidisciplinary and it's involved many people over the last few years. Essentially, it involves a lot of manual annotation of data. So these are some examples of anonymised little extracts, reported fleeting suicidal thoughts, strongly denied acting on suicidal thoughts, little phrases from the texts of electronic health records. And of course, because in our use case, we really want to prioritise picking up any mention of suicidality in the affirmative, We had to make rules to decide whether a patient overall had ever had a mention of suicidality, in which case they would be considered a positive case of suicidality, or whether they just hadn't in a particular document. This is just one example. Another interesting piece of work that we did was to look at how language changed between times when a person was healthy and the times for that same person when they were in 30 days prior to a suicide attempt. And we have shown here a couple of schemas, a healthy tag cloud in a healthy period and a suicidal tag cloud, and have analysed the differences between those times. I just want to bring you on finally to another piece of work that I'm moving on to with my team, where we're looking at data science to begin to understand how social media and smartphone use may relate to self-harm behaviour. And I think this is a really important question in the digital age. This work's being sponsored by the MRC and the Medical Research Foundation, and we're in partnership with the national charity Young Minds. Every week we all see news headlines focusing on the dangers of social media, the potential harm to mental health from apps such as Instagram. But are they really so perilous? We do not yet have the hard facts. Smartphones and social media are both here to stay, and for young people in particular, it's part of their lives. But I think throughout the generations, we're going to find that technology is there and we can harness it for good. And that's my real interest as a psychiatrist. We're working on this consented study of 1 year duration to look at how people who are vulnerable to self-harm, so they're known to mental health services, how does their use of social media vary, their linguistic patterns, the timings of their posts, different apps, what themes they focus on, is their nighttime use any different to their daytime use, and is there a safe limit? I think this is crucial work because at the moment, our professional clinical bodies, such as the Royal College of Psychiatrists and British Psychological Society, suggest that clinicians should routinely inquire about social media activity, internet use and online gaming as part of the assessment of young people. However, no one knows how this should be done, let alone if it adds anything to the assessment. So thank you very much. I realise I'm standing between you and coffee, and I think Paolo's come up to tell me that. So thank you for your attention and do enjoy your coffee break.
Thank you very much for giving the time as well. Yes, indeed, we have reached the coffee break. It's been a very interesting first part of the festival. I think we are running 5 minutes or so late, so we are probably starting 5 minutes later than scheduled. So I think 5 minutes to 4 we will be back for the second part of this research festival.
Hello everyone, welcome back to this second session of this year's IoPPN Research Festival. I'm very pleased to introduce our next keynote speaker. It's Professor Temi Moffat, who's Professor of Social Development at the SGDP. She is very kindly recorded a lecture for us. We'll start shortly.
Hello. I'm sorry we can't be together for this year's research festival, but we're just going to do the best we can. I'll tell you about surprises from charting 4 decades of mental disorder from childhood to midlife. Let's begin here. We clinicians and mental health researchers tend to see a patient at only one point in their lives. That's when they come to us for treatment or when they sign up to take part in one of our studies. We make heroic efforts to diagnose the presenting condition correctly at that time. And we need to ask ourselves, why do we do this? Well, we do it because our thinking is that their diagnosis adequately represents their psychopathology. And also, it tells us what might have caused it and what treatment to choose and what their prognosis will be. Here you see 3 patients, each with their own diagnosis, or in the case of the person on the right, a pair of comorbid diagnoses. This slide contrasts the typical cross-sectional way of seeing the diagnosis of mental disorder against a different view, following people through time. Our team has had the rare privilege and opportunity to follow the same 1,000 individuals for almost 5 decades, and we noticed something that surprised us. The bottom picture shows you this. We found that people in our longitudinal study changed their diagnosis very often, and that is what this talk is about. First, I need to tell you about the study. It takes place in New Zealand, in a city called Dunedin, which is down at the bottom of the world. Here I show you the design of the Dunedin study. If you look at the top left, you can see that the study began in 1972 with all the babies born in one city. And down the left-hand side, you see the ages at which each study member came into our clinic for a full day of assessment. And down at the bottom, you see that we most recently followed them up at age 45, finishing in 2019 when 94% of those still alive took part. The Dunedin study is by now the longest running study of mental disorders. It has survived 4 editions of the American Psychiatric Association's DSM. In fact, it was the first study to use a standardized diagnostic interview to assess mental disorder in children at age 11 in 1983. This shows you the ages of the study assessments over each version of the DSM. The ages range from 11 to 45. During that period, we carried out 9 successive standardized diagnostic interviews. Here's the list of 17 DSM disorders that are assessed in the Dunedin study. They're organized for you by family, with externalizing on the left, which contains ADHD, conduct disorder, and substance abuse disorders. Internalizing disorders are in the middle, containing depression and different kinds of anxiety disorders primarily. And then on the right are thought disorders family, with schizophrenia and mania, and you see there OCD, included because it was considered by the DSM-5 for a thought disorder, and factor analyses carried out at that time in multiple different data sets showed that OCD symptoms do fit with mania and schizophrenia. Now, clinicians interviewed each Dunedin study member using standardized diagnostic interviews. To get a diagnosis, the study member had to meet the full DSM criteria, and we also required self-rated impairment, and we further documented corroboration of key symptoms by informants. Here we ask 3 overarching questions of these data. Who gets a mental disorder? When do they get it? And do people retain the same diagnosis over decades of their lives? I'm showing you the most surprising finding first. Across 4 decades of life, cohort members moved in and out of internalizing, externalizing, and thought disorders. Down at the bottom, you see in black when a study member had died. In gray, times when a study member was missing at one of the assessments. At the top, you see in green periods when a study member was well and had no mental disorder. To represent the disorders, blue is for internalizing, violet is for externalizing, and orange is for the thought disorder family. Blended colors represent times when people had multiple families of disorder. For example, dark purple shows people who had both internalizing and externalizing at the same time. This is just a quick glimpse to get you thinking, and I'm going to unpack the data now, and then we will come back to this chart later. So how many people experienced a mental disorder in the first half of the life course? 85% of the Dunedin cohort had onset of disorder by age 45. You can see this in the bar on the left. On the right, you see the past year prevalence, and it looks as if it's going down somewhat at the older ages. Part of this is because people with mental disorders tend to die young from suicide, overdoses, accidents, and diseases as well. Now, you've probably been wondering, is the Dunedin cohort the only study to show this high lifetime prevalence rate of disorder? Is there something terribly, terribly wrong with this study? Let's begin by looking at the treatment data. Here you see data from the Danish National Treatment Register. There's data for 6 million men and 6 million women, and the chart shows that 35% of the Danish population has been treated for a mental disorder, whether in primary care, as an outpatient, or in a psychiatric hospital. But you know that most people who have disorder don't ever get treated, and so this is an undercount. So we move to the national comorbidity surveys, where you see that 45 to 50% lifetime prevalence is showing. But these surveys rely on retrospective recall by older respondents, and many respondents forget that they had symptoms of disorder some time ago in their younger days. So this too has got to be an undercount. Here you see data from 5 longitudinal cohort studies that have carried out repeated diagnostic interviews located in the United States, Switzerland, and New Zealand. These 5 studies counted up the lifetime prevalence of mental disorder as it accumulates going forward. Cohorts report lifetime prevalence of total disorder somewhere between 65% and 85%, which looks pretty similar to that in the Dunedin study. Higher lifetime prevalence has been reported from studies that started the youngest and had the most waves of assessment, and this makes sense. So the Dunedin study estimate of lifetime prevalence of mental disorder is not an outlier, and it is probably correct. If you stay mentally well your entire life, you are not normal. So what are the implications for the public's understanding of mental illness? First, mental disorder affects almost everyone eventually. Skeptics claim that this is an excellent example of how diagnostic expansiveness runs wild and pathologizes everyday life, and that the DSM medicalizes the normal ups and downs of life. However, I'd like you to recall that all the diagnoses in our study involved impaired functioning, and they were corroborated by informants. And many people with mental disorder do absorb healthcare resources. Also, high prevalence does not necessarily diminish significance. If it did, we'd have to say, because of the obesity epidemic, that obesity has little consequence for people's health. Our data suggests that some mental disorders become chronic or recurrent, like diabetes or COPD or Parkinson's disease. Other mental disorders resemble a fractured leg or early-stage breast cancer or viral respiratory infection. They're disabling, but relatively short-lived and curable. We hope that if the public realized that sooner or later everyone has a brush with mental health problems, this recognition would reduce stigma against mental illness. I like to say, if you haven't had your mental disorder yet, it's probably coming to you. And less stigma ought to promote more people to get treatment and to get it sooner. At what age do most people experience their first disorder? In Dunedin, about 35% were diagnosed by the study by the age of 15. You see that on the left. And then another 25% were first diagnosed at the age of 18, totaling to about 60% of the population who has onset of mental disorder as a child or adolescent. Is early onset of disorder a harbinger of more disorder to come? This chart shows early onset predicts more subsequent years with disorder, lifetime. Each column is a wave of the study. Darker purple color shows that more study waves with a diagnosis. On the left, you see study members with the earliest onset tended to have disorder at 4, 5, 6, or 7 subsequent study waves. This chart shows that study members with early onset also tended to experience more different types of diagnoses, diversified over their lifetime. Here, the darker purple color equals more diagnoses. On the left, you see those with onset as a young person tended to have 3, 4, 5, or even more different kinds of diagnoses made by the study during their lives. Here we see how the diversity of comorbidity accumulates over the life course. I want to show you how it builds up. So I'm going to start with the data from aged 11 to 15 years, where you can see that about 35% of people had a disorder, and almost half of those had 2 disorders at the same time, and almost half of those had 3 disorders at the same time. You can watch as more people get diagnosed and more people experience multiple different disorders. Age 11 to 18, 11 to 21, 11 to 26, 11 to 32, 11 to 38, and 11 to 45. By age 45, over 80% of people who experienced one psychiatric disorder had experienced at least one other psychiatric disorder. At this point, it became clear to our research team that 3 measurement parameters were emerging that can define and describe a person's mental disorder history pretty well: age of onset, length of duration, and extent of comorbid diversity. These 3 parameters are highly correlated with each other in the Dunedin cohort, with Pearson correlations somewhere between 0.6 and 0.8. You might be thinking, well, sure, if the onset is very young, there's simply more time for duration and comorbidity. But these associations remained after correction for the number of years available for observation between each participant's age of onset and the end of the observation period at age 45. The important point here is that some people will have a mental health history that starts young, lasts a long time, and includes many different disorders. Other people will have a lifetime mental health history that starts later in life, comprises only a brief episode, and just one diagnosis. But most people will have a pattern somewhere in between. Does anyone retain just one exclusive diagnosis over their lifetime? Do people tend to stay within a disorder family over their lives? This is an important pragmatic question because so much of our work as mental health professionals is diagnosis driven. We have specialist clinics for depression, ADHD, substance abuse, psychosis, eating disorders, and so on. We have treatment protocols that are tailored to each specific diagnosis. We have journals that are disorder specific, and in the United States, there are even funding agencies that are disorder specific. This chart shows that in the Dunedin cohort members who ever had disorder, it was rare for a person to retain one single diagnosis type over their lifetime. If you look on the left, on the blue column, this shows those who ever had any internalizing disorder. At the top, The lightest blue part shows those who kept their specific diagnosis. They only ever had major depression or only ever had panic disorder, for example. There were only 12.7%. The medium blue shows 18% had comorbid diagnoses, but they stayed within the internalizing family, some combination of anxiety disorders or anxiety and depression. But the darkest blue shows that 69% of individuals with an internalizing disorder also had diagnoses that went outside the internalizing family. They developed an externalizing disorder or a thought disorder. This was similar for people who had an externalizing disorder. The violet column shows you that 75% of them also had an internalizing or a thought disorder in the 4 decades of the study. And the orange column shows you that all but 1.8% of individuals with a thought disorder had disorders in other families as well, outside the thought disorder family. Do people experience different kinds of psychiatric disorders across their life course in some kind of meaningful sequence or pattern? You saw this chart before. Over 4 decades, people had lots of different disorders at different times, and they moved between types of disorders in patterns that looked nearly random. The data point to something like a hydraulic process. Maybe there's a general vulnerability for psychopathology, and it flows through the life course like an underground river, and at different times it bubbles up to the surface. But each time it takes the form of qualitatively different symptom pictures. We traced 1,037 study members for 35 years and found 692 patterns of diagnosis sequences. 599 of these were unique. It was rare for any 2 people to follow the same sequence. But maybe this huge comorbidity happened just because we were interviewing people in the community. Does it really apply to the patients that you see in clinical settings? There are 70 study members in the Dunedin cohort who have been treated as an inpatient in hospital for mental disorder or substance dependence. This picture means that serious cases who are found in clinical settings are highly likely to cross disorder families when followed up later in their lives. We had traced these 70 cohort members who were hospital inpatients for the 35 years of the study, and we found 70 patterns of diagnosis. 70 of them were unique. No 2 patients followed the same sequence. This finding is not limited to New Zealand. Something similar was also reported last year from Denmark's nationwide electronic medical registers of treatment for mental disorders. The paper appeared in JAMA Psychiatry. It concluded that over a 10-year period, among several million Danish citizens, people switched diagnosis often, and every mental disorder was associated with significantly increased risk of every other mental disorder. So what are the implications of our findings for clinicians? First, disorder typically onsets by adolescence, and if so, it persists for decades thereafter. So it's really important to treat young people and treat them effectively. Next, very few adults develop mental disorder for the first time as adults in midlife. Your adult patient probably had different disorders in the past, even if they don't remember them. And they will have still other different disorders in the future. This means treatments cannot just address the current presenting disorder, but must also build a patient's skills for maintaining enduring mental health years into the future. Of course, we have to treat the presenting symptoms, but also, how can you help your patient ward off their next disorder? By teaching basic skills that can prevent all kinds of disorders. In view of this, research and clinical work that focus only on a presenting diagnosis look pretty short-sighted. And yet, as I noted before, Our field continues to focus our attention one disorder at a time. I reviewed the table of contents in JAMA Psychiatry, American Journal of Psychiatry, Lancet Psychiatry, and Molecular Psychiatry last month in preparation for this talk, and I found that over 75% of the articles published there were on only one diagnosis. Thank you for joining our research festival. Please write to me at the email address that you see here if you have comments and questions about this talk today. And again, thank you for joining the IoPPN Research Festival 2021.
Thank you for that brilliant from Professor Moffat. If you want to hear more about her work, she's been interviewed by the Mental Elf, who's also tweeting about this event, and that's available on the National Elf Service SoundCloud, where you'll also find an interview with the first keynote speaker, Professor McAlonan. So I now hand over to our next speaker, who is Dr. Rosalind Moran, and she's a reader in theoretical neurobiology.
Thank you, Mittel, and to the large crowd for having us here at this exciting afternoon. What I'm going to do in the next few minutes is take you on a small whistle-stop tour of something called computational psychiatry, and take a little look at some neuromodulators and how we might understand also normative aging and some of the decline seen in cognition in aging, since our theme is across the lifespan using computational tools and tooling. So computational psychiatry is a fairly nascent field, though it's now a strategic mission of the National Institute of Mental Health in Bethesda under Josh Gordon. And in this book that we put together at an Ernst Strungman forum, it was an appeal for modelers to come together with psychiatrists to try and go a little bit beyond biological psychiatry and more formally describe what we think thought process is and the biological process that supports it. So effectively what we're trying to describe is both normative theories of the brain, or what the goals of a brain may be trying to do, context dependent or not, and also how the cogs and wheels work towards those particular goals. And so a common frame and a fairly simple but interesting mathematical frame, and one that you've seen in AI situations play Go, is that of reinforcement learning and its putative explanation of midbrain dopaminergic and potentially also serotonergic modulations. And so just in this first study, I'm just going to say that neuromodulators are important. We don't have to be told that. This is a paraphrasing of a quote that our drives, desires and pursuits are controlled by our neuromodulators. Everything else is a lookup table, has been ascribed or credited to many people from William James to Tom Poggio. But we know that these are exquisitely small numbers of neurons and exquisitely important. And ironically, what we did was try and look at residual dopamine in people with some disorders of dopamine, and also in patients with Parkinson's, and also in patients with essential tremor who were undergoing deep brain stimulation surgery at Wake Forest University in North Carolina. And here the surgeons bravely, and the patients bravely, allowed us to put carbon fibers down the bend gun array where their DBS electrode was going to be placed and take measurements known as fast scan cyclic photometry from the striatum while they played a market investment game. And this offered the first fast transient measurements of serotonin and dopamine in humans at a timescale that was aligned with specific behaviors. And so we first took a look at dopamine, and we know it's classically associated with these so-called reward prediction errors. And we were able to look at the concentrations after sifting and modeling these data from fast-scan cyclic photometry, showing indeed that from 21 patients who participated in the task, we could see significant effects of positive and negative reward prediction errors, creating positive transients and dips in the dopaminergic output at the striatum, as was expected from the theory. But what we were particularly interested in was looking at serotonin in the same region while these people played this neuroeconomic game. And we know that we're prescribing SSRIs successfully in depression, but where serotonin is more of an elusive neurotransmitter, neuromodulator, and there are many theories on what it does, from learning in negative context to patience, as in waiting for some outcome. And so what we did was look at serotonin in the context of how people change their mind or change their behavior after a bad event. And so what we did was look at how a previous bet was then altered because they were moving these buttons. And so we could ask what, for example, if something bad had happened, if things were worse than expected and people made less risky bets on the next game, what we found was that serotonin promoted this sort of behavior to become less risky. And then if we looked at the other end of the spectrum, when people are very risk averse and asked how serotonin impacted behavioral change, what we saw in these tasks was that in order to be riskier, serotonin actually supported or gave us positive fluctuations where we were in these low regimes. So effectively, serotonin was associated with both approach and avoidance, or trying to get you to a sort of safer middle ground that would be promoting you away from being too risky or too risk averse, and what we call these were protective action encoding of the serotonergic transients in the midbrain. So reward and how we respond to things in the environment in a rewarding way is certainly one ubiquitous and dominant theory in computational neuroscience generally, and in the field of computational psychiatry. Another one that my lab look at is known as the free energy principle. So this is what some philosophers have called a desert earth theory, a tautology, or something that almost has to be true. And it says that our brains try and minimize a computable bound on surprise, which is technically known as the free energy. And if we unfurl the mathematics, which I won't do in these 5 minutes, what you see is that some of the clues in the neurobiological architecture that we know to be present in our brains, for example, its hierarchical structure. This is a picture from Feldman and Vanessen's classic macaque wiring diagram, but also these so-called canonical microcircuits, so circuitry that seems to repeat, particularly in the sensory cortices, might have a mathematical bent that helps us minimize this surprise and do this mathematical computation. And you can boil it down after some mathematics to a few little corollaries. One would be that backward connections coming top down to lower levels in the hierarchy might be providing predictions, while forward connections up our cortical hierarchies might be signaling prediction errors. And this is an interesting way to look at beliefs in the context of aging, as I'll show you in a second, but also in the context of non-traditional neuromodulators like the microbiome. So I'm jumping a little, but just to show you that what we're studying in this is a fear conditioning task, and we're trying to understand how people respond in this hierarchical way of forward and backward connections when they're presented with this CS plus, so this shocking stimulus, as opposed to the CS minus. And we're trying to associate it with alpha diversity in the microbiome, which is a metric of microbiota and its genetic composition that's been associated with positive promotions in mental health, like less anxiety and depression. And so just generally in the task, what we saw was that some usual suspects showed up in this fear or threat-like paradigm, including the anterior insula and the anterior cingulate cortex. And we took a look at the connections in this network while people are processing this potentially fearful stimulus in the context of the connections between the anterior cingulate and the anterior insular cortex using a so-called dynamicism model for fMRI. And what we found was that we have a correlation, again, no causation here, but a correlation between how much top-down negation you have from the anterior cingulate cortex to the anterior insula that's associated with an individual's alpha diversity within their microbiome. And if we look at our computational framing, what this top-down connection from the ACC to the insular cortex is doing is potentially shutting down or explaining away, as we call it in the literature, the prediction errors that might arise when you get a fearful stimulus or even just the presence of the stimulus. And this top-down or bottom-up processing, we've also taken a look in the context of fMRI again, with this is work with Suki Shergill, looking at how we might see this model of top-down and bottom-up changes in the context of treatment-responsive schizophrenia, as opposed to patients who don't respond to dopaminergic medications. And here, interestingly, what we've shown was that, again, cingulate back to sensory cortices was switched on or enabled in those patients who were going to respond or had responded to dopaminergic medications, but were entirely absent in patients who were treatment resistant. And so this distinction between bottom-up and top-down connectivity in the brain is perhaps most relevant in this lifespan conference in terms of looking at aging brains. And you can imagine, because we might get older, we might have more predictions as we get older, and our predominant direction of travel might be top down rather than bottom up. And we've shown that in some studies using electrophysiology, where we've looked at just simple priming tasks where you're trying to name and rename items on a screen. And what we did was look at the direction of signaling in younger and older brains, and found indeed that we might have some basic inputs into frontal regions that are controlling or orchestrating the response to the visual input in the older brain, but not in the younger brain. And indeed, if you look around the frontal cortices, you can see that the older frontal evoked response shows that you have a faster frontal onset than in the younger cohort. And this is associated with positive performance improvements in the older cohorts, so faster reaction times in the priming task. And we've also shown it in the context of sensory learning paradigms, that what this might mean, again, in a model analogy, is that older brains are less brittle, they're better contextualizers, and that they have more top-down control on incoming sensory information. It also explains some of the literature that you've seen in the classic normative aging, in particularly neuroimaging, where you see things like de-differentiation and hemispheric asymmetry reductions as you get older. But we interpret it in a sanguine way as a model that's more generalizable. So literally, as you get old, you don't overfit your sensory data. However, there is no free lunch. So looking at explicit recall or memory tasks that require medial temporal lobe proper, so regions of the hippocampus, to do the recall, what we found here is that the the top-down predominance from, in this case, regions of the inferior frontal gyrus back to the hippocampal cortex is associated with impairment in memory recollection at an inter-individual level. So while we may be having generalizable models that we can apply in different situations as we get older, that's informed through this theoretical understanding, we also have evidence, unfortunately, of some other things that we may lose. And with that, I'll thank you for your
you are out of time. Perfect.
Am I on time? Very good.
Yeah, you're slightly over. That's okay.
Sorry.
I'll interrupt you, but thank you very much.
There we go.
I'm just going to invite our next speaker. We're a little behind, so I'll just move us swiftly on between our speakers. Our next speaker is Dr. Gemma Modinos, who's a reader in neuroscience and mental health, but she's also a Sir Henry Dale Fellow at the IoPPN.
Yes, thank you, Mitul, for the introduction. I'm delighted to speak today at the IoPPN Research Festival. I'm Gemma Modinos. I'm a reader across the departments of psychosis studies and neuroimaging. And today I will give you a bite-sized overview of the work that we do in the lab. Our overall aim is to try and understand the role that emotion-related brain systems may play in the development of psychotic symptoms or psychotic disorders like schizophrenia, and whether targeting these mechanisms early could be a new way to try and prevent or delay the onset of psychosis. So I'd like to start by thanking the fantastic group members in my lab, our funders, as well as the wonderful collaborators across the IoPPN and the clinical services that we work with. Now, the first question, what are the neural bases of emotion across a psychosis continuum? So one way in which we can answer this question is by using brain imaging, and we have a participant in the scanner and we can ask them to do tasks that elicit emotion responses or that we ask them to identify emotions. And of course, for this, you need to have a good range of emotional stimuli, and things like Chuck Norris wouldn't really work very well. What we did with this work is we found that people from the general population with subclinical psychotic-like experiences or people with a clinical high risk of psychosis, they show increased reactivity of emotion-related brain regions during emotional processing. And recently, with the UGEI multi-center dataset, we were able to study 265 individuals. And we found that clinical high-risk individuals who had increased responsivity to negative emotion at baseline, this was associated with reduced gray matter volume in key emotion processing regions such as the hippocampus and the ventromedial prefrontal cortex. And this was particularly associated with the development of a poor functional outcome at 12 months. And I would like to point you as well to Paulina Lucco's meta-analysis. She's a PhD student in the lab. She's meta-analyzed this evidence on fMRI of emotion processing in early psychosis. And something I'm also excited to tell you about is that thanks to the work on emotion, we have an art science collaboration funded by King's Artist Award. And we have an artist in residence, Izzy Parker, with whom we are doing a series of workshops with youth groups and also with service users. Now, the next question, what are the neurochemical bases of this increased responsivity to emotion? This is important when we're thinking about new treatments. And we've been focusing on excitation-inhibition balance. So this is a process by which your brain helps keep an optimal level of brain activity. And what we do is we apply a cross-species translational imaging approach. So we are using the same imaging modalities or comparable imaging modalities across our humans and animal models. And this is an example of our work imaging the GABAergic neurotransmitter system. The work with animals, with a neurodevelopmental animal model, is in collaboration with Professor Tony Grace's lab at the University of Pittsburgh and is currently being prepared for publication. And we've also had a great collaboration with Tony Vernon's lab here at the IoPPN, which we found that antipsychotic exposure modulated the density of specific GABA-A receptors in this corticolimbic circuitry. We also use comparable imaging methods with other animal models. For example, a more precise genetic animal model of disruption in a particular type of GABAergic cells could express the protein parvalbumin. And as you can see here, we can use comparable imaging methods to quantify levels of the neurotransmitter glutamate across mice and human. And we can also measure cerebral blood flow across mice and human and resting state fMRI connectivity. And this is also currently being prepared for publication, and it's a collaboration with the RICO and Marine Labs at Guy's campus. The human studies, as you can imagine, they were severely impacted by the COVID-19 pandemic shutdown, so it will take a little bit longer to have those out. And finally, what we also do in the lab is we complement these really small, bespoke, precise mechanistic studies with a large scale project called the Enigma Schizotypy Consortium, which I organized and I lead since 2018. We currently have 29 sites worldwide, and this is a network of neuroimaging researchers worldwide trying to understand neurobiological bases of psychotic-like experiences in the general population. Our first paper is now in preprint, and what we found is that in 3,000 scans is the greater levels of psychotic-like experiences, the greater the cortical thickness in this region you see here in red, which is the ventromedial prefrontal cortex, and is a key region in emotion processing. And finally, what we've also done is to correlate the effect size map of cortical thickness effects in schizotypy with the effect size map of cortical thickness abnormalities from the Enigma Schizophrenia Consortium. And we found a significant positive correlation between these effect size maps, which wasn't present between schizotypy and bipolar disorder or between schizotypy and major depression. So really supporting a continuum view of psychosis from psychotic-like experience to then more clinical expressions like schizophrenia. So this is all from me. Thank you very much for your attention. I'm happy to take any questions in the Q&A.
Thank you, Gemma. That's amazing work. I will now hand over to our next speaker, who's Professor Jerome Breen, Professor of Psychiatric Genetics here at the IoPPN SGDP. Jerome.
Thank you, and thanks to all the speakers for excellent talks so far, but I hope I can keep it up. It's a pleasure to talk to you today about eating disorders. So first of all, to say that we regard them in our research as being both genetic and environmental in their cause. So they affect about 5% of the population, but there's a strong female predominance such that maybe 8 or 9% of females may develop an eating disorder at some point during their life. Unfortunately, eating disorders are also the least treated and least well researched in psychiatry, with the worst outcomes, including very high mortality rates. However, they are also 50 to 60% heritable, which is much greater than the heritabilities we observe for depression or anxiety. And I think they have the most interesting biology of any set of psychiatric disorders. Why do I think this? Well, for the last 4 years, I've had the pleasure of co-chairing the Psychiatric Genomics Consortium Eating Disorders Work Group. In 2018, we published a study of nearly 17,000 cases of anorexia nervosa. And we found 8 genetic variants associated with increased risk, including 4 single genes with very interesting biology. However, we also found evidence that suggested anorexia was not just a psychiatric disorder, but rather was a metapsychiatric disorder. The primary analysis method from which we concluded this came from genetic correlation analysis. This analysis looked at the genetic similarity between anorexia and a range of outcomes, or the genetic correlation. First of all, we found that anorexia had strong positive genetic correlations with psychiatric disorders. This is as we would expect. The genetics of anorexia nervosa clearly overlaps that of other psychiatric disorders. However, we then found that anorexia had negative genetic correlations with a range of metabolic parameters, including insulin resistance and hormones such as leptin, as well as a range of body composition parameters, including body fat percentage and obesity. Negative genetic correlations imply that anorexia nervosa cases as a group had lower germline genetic predispositions to having high levels of these traits, or in other words, have the genetics of low levels of these traits. We concluded that from this data, genetically, anorexia nervosa is both a metabolic and psychiatric disorder. Now, we want to accomplish this type of study in every eating disorder. Anorexia accounts for only 1/5 of eating disorders, but we lack the sample sizes required to do this. So I and others have created an initiative called the Eating Disorders Genetics Initiative. This is led by Cindy Bulick internationally, and I lead the UK arm of this. In the UK, we work with the charity Beat, and we want to recruit 10,000 people with experience of an eating disorder to complete an online questionnaire and to give us a saliva DNA sample. In doing this, we want to create a platform for faster and easier research in eating disorders. Unfortunately, we launched our EduUK project on the 26th of February 2020, which pretty much marks the start of the pandemic in the UK. This threw a bit of a spanner in the works, but we have nevertheless managed to now fully recruit 1,500 individuals, and over 4,000 people have answered our questionnaires. We've already begun to analyze the data, and at the moment we're asking relatively simple questions, but ones which are clinically important, such as what is the age at onset distribution of eating disorders in our sample? We find that only 1/3 of our cases have reported an onset of eating disorder at below the age of 16. This is important because most clinical services for eating disorders in the UK are targeted at children. Instead, we find that actually more than 90% of cases report onset below the age of 26. And this has led us to hypothesize that the neglected majority of new onset eating disorder cases could actually be treated if there were additional services for eating disorders targeting ages 16 to 30. I would just like to express my gratitude to staff working the project, including Dina Monson and Helena Davies, pictured here. If you want to find out more about our research, go to edgyuk.org, our website, and you can contact us that way as well. Thank you.
Thank you, Jerome. Fascinating insights from those large data sets. Great. Our next speaker is Dr. Maria Eliso Serrano Navarrete. I hope I pronounced that correctly. And she's a research associate in neuroimaging. Over to you.
Thank you. Thank you. Good afternoon. And thanks for this opportunity to present some of my research that I did back in Belgium at the University of Liège about changes in synaptic density across the lifespan. So humans are constantly growing, changing, learning, and the brain, the principal controller of our bodies, is also in continuous change. This organ is composed by billions of neurons which communicate each other by synapses. And these synapses are created and eliminated in response to internal and external stimuli. And the interest for quantifying them has always been present because it would help us understand how the brain develops and the emergence of neurological diseases. However, it was not until the development of the so-called synaptic density tracers, that are SV2A radio tracers, that we could measure these changes in vivo. So basically, these radio tracers bind to the SV2A protein, which is a presynaptic protein present in the synaptic vesicle, whose role is allow the neurotransmitter release, and therefore, I'm going to put the pointer, so yeah. And so it's involved in the neurotransmitter release. So therefore, is involved in the process of the synaptic transmission, so the process in which neurons communicate each other, the information. So during my recent research, I used this radio tracer and I measured in vivo, thanks to the positron emission tomography, changes of synaptic density through the lifespan, through the brain, rat brain development from puberty to adulthood. For you to better understand, in this picture, we can see in yellow regions that have an increase in SV2A concentration, and in red or dark colors, regions that have a decrease or a low concentration of SV2A. So as you can see, through the development of the rat brain, there is an increase in SV2A concentration in the brain. So when you quantify these images, you can observe that this increase is not only present in the whole brain, but is also present in different regions, such as the hippocampus, that is a region important because of the implications with memory process. This increase in SV2A can be related to an increase in synaptic density, but also to the strengthening of the synaptic connectivity between neurons. And this topic has been and is currently being explored by the University of Edinburgh, in which they are comparing, for example, males and females. But, well, the interest of my research was not only to analyze how the brain develops in a healthy condition, which is very interesting, but also to know how this development is affected by a pathology. In this specific case, I studied epileptic animals. So, as you can see in the images, these epileptic animals present also the increase in SV2A concentration, so the increase in synaptic density through the development of their brains. But this increase in synaptic density is affected by the epileptic condition. So when I quantified these differences between groups, I could see that there were significant differences between control and epileptic animals. So the epileptic animals presented lower synaptic density in different brain regions and in the whole brain. And these significant differences were very interesting because they were present even slightly before the onset of the seizures, so when they were young adult and during the latent phase. So currently, this synaptic density loss is being studied in vivo in multiple neurodegenerative and neuropsychiatric diseases with SV2A radiotracers. And well, even if there is a lot of validation to do and a lot of research to do, the results point out that the SV2A radiotracer are a promising tool to study changes in the brain development, and also they are promising biomarkers for diagnose different neurodegenerative and neurological disorders. So thanks a lot for this opportunity, and if you have any questions, please ask me. Thanks.
Thank you very much, Maria. That's brilliant. We're in the same department, but we'll get to meet properly when we go back. So I'm going to hand over to Dr. Virginia Carter-Leno, who is a Sir Henry Wellcome Fellow. Are your slides up?
1 second.
No worries. Ginny is from the Department of Biostats and Health Informatics.
Yes.
Fantastic.
I've moved over from child psychiatry.
She's moved over from child psychiatry. I should be talking about the associations between theory of mind. Oh, it's up. Okay, it's up. I won't give the title. I'll hand over all yours.
Okay, thank you. Sorry about that. So yes, thank you for inviting me to talk about a piece of work looking at associations between theory of mind and conduct problems in autistic and non-autistic youth. So this work was motivated by research coming out, which has demonstrated that the prevalence of co-occurring psychiatric difficulties or mental health difficulties is much higher in autistic individuals. And you can see this study here from Professor Simonoff in the Child and Adolescent Psychiatry Department at King's, which showed that in a population representative sample of 10 to 14-year-old autistic children, 70% met diagnostic criteria for an additional psychiatric diagnosis. And you can see the kind of highest ones were conduct or oppositional defiant disorder, emotional disorders like anxiety and depression, and ADHD. And these prevalence rates are substantially higher than what we would expect in or compared to typically developing children of the same age. And so that then raises the question as to why the prevalence of mental health difficulties is so much higher in autistic individuals than autistic children. And so there are different ways to think about this in terms of hypotheses and testable theories. So this is kind of one way that I've been thinking about it. So in typically developing populations, you have established risk factors, and these can be intrinsic or extrinsic, so kind of exposures, things that happen to you, or individual traits. And if you have these risk factors, then you're more likely to go on to develop mental health problems. And so one hypothesis is that in autistic people, it could be that these risk factors are more prevalent. And this then bumps up the rate of mental health problems and can explain the increased prevalence. But an alternative hypothesis is actually that the risk factors are not more prevalent, they just for some reason have a stronger effect. So, you know, given being exposed to the same risk factor, an autistic person is more likely to go on to develop mental health problems. So we wanted to use some data to try and test out these two theoretical models and see which one was better supported. So we started out thinking about risk factors that we are kind of known to be associated with mental health problems that might be especially pertinent or relevant for autistic children. And so we settled on theory of mind. So this is the kind of social cognitive ability to infer and understand the mental states of others. And in typically developing children, this has been fairly robustly associated with conduct problems. So I'll just use conduct problems to kind of cover all manner of externalizing symptoms, but just for simplicity here. And theory of mind difficulties are, on average, thought to also be present in autistic youth. And so, but no one had actually looked at, well, in autistic individuals, do theory of mind difficulties also predict behavior problems in the way that has been demonstrated in typically developing youth? So this is what we set out to do. So we combined data from two well-characterized cohort studies, which gave us 128 autistic youth aged 10 to 16, and then 50 non-autistic comparison kind of counterparts. And I call them non-autistic because about half of the sample had a diagnosis of mild intellectual disability, and then the other half were taken from mainstream schools nearby. So we asked parents to fill out the strengths and difficulties questionnaire to measure their child's conduct problems, and then we asked the young people themselves to complete this measure of kind of subjective mentalizing or theory of mind called the animated triangles task. And so the first thing we did was look for group differences. So we would expect that the autistic children would score lower on this task. And this is indeed what we found. So the two metrics you can kind of pull from this task, the non-autistic group is scoring higher. But then the key question is, and how do theory of mind difficulties relate to conduct problems, and are there differential associations between the autistic and the non-autistic group? So kind of statistically speaking, does autism moderate the association between theory of mind difficulties and conduct problems? And so we actually found that very clearly it did not. So there was a negative association between theory of mind ability and conduct problems regardless of diagnostic status. And we also looked at some other mental health outcomes that I kind of alluded to in the beginning of the talk, which are of interest in autistic populations, so ADHD type symptoms and emotional problems, and found pretty much the same thing. So to conclude, the strength of association was comparable between the autistic and the non-autistic children, which would support the greater prevalence hypothesis. But also I think the results suggest that theory of mind difficulties are often kind of archetypally thought to contribute to the core symptoms of autism, but actually they may play a role for co-occurring mental health problems. And if we can identify what's driving co-occurring mental health problems, then we can put support and intervention in place and promote positive mental health for all autistic people. So thank you for listening, and I'm happy to take any questions.
Great, thanks, Jenny. If you've got some questions, you can pop them in the chat, in the Q&A section, and then she'll be able to answer by text. Fantastic. So our next speaker is Dr. Sam Cook, who's a senior lecturer in neural plasticity. Are you ready, Sam?
Yes, hello, Mitchell, thank you. Hopefully you can see my presentation.
Yep.
Good. Okay, so let's get into presentation mode. What I'm going to talk to you about today is learning and memory, and this is obviously a very relevant topic to the theme of across the lifespan, because cortical plasticity in particular is incredibly involved in developmental processes, but also we start to see deficiencies in that process as we age. And one of the key things when it comes to studying learning and memory, which has been the focus of my career, is to be able to study the biological underpinnings of that, the mechanisms that occur in the brain as an animal learns. And this has been quite a tricky thing to do. And one of the attempts that we've made to solve this problem is to study a very fundamental but quite tractable form of learning and memory that's really, really critically important for all species of animal. Okay, so what you're looking at here is a baby, a very young baby, an infant, looking at a very simple visual stimulus and then undergoing the process of what's called habituation. So this is basically the process by which you learn to ignore all of the unimportant things in your environment, and you'd be surprised to know perhaps that learning what's unimportant is really critical. Okay, so this is a way of filtering out all of those things that could drain our energy and attention and allowing us to focus on the really fundamental things that we need to understand about the environment. So you can see this baby undergoing very profound habituation here. It's initially very interested in the stimulus, but that interest very swiftly wanes. And then you can do a test where you simply interleave presentations of that familiar, now highly familiar orientation. You can see the infant's not even looking at it. And all you have to do to re-energize the infant's interest in the stimulus is to change the orientation of the stimulus. And you can see that here. Suddenly, it's just as interesting as it was the first time. And we'll probably all have experienced something along these lines with children, but we do it ourselves all the time. And in fact, as we get older, not only do we undergo this short-term habituation, this adaptive filtration during a single session, but we carry savings from that over to the next session. So this becomes a long-term memory of things we're familiar with. Now, we study this process in mice, so we do a very similar thing to the mice that you see here with the baby. We show them these simple visual stimuli. And you can see here that the animals are fixed, head fixed, they're comfortable but head fixed in front of the stimulus. And this allows us to record behavior, in this case just simply the movement of the animal to a particular stimulus being shown, while also recording the events that are occurring in this case, the neocortex, the primary visual cortex of the mouse. And you can see, first of all, here a very similar effect to in the baby, where the animal undergoes habituation as that stimulus is repeatedly presented on the screen. You can see here that the behavioral response is reduced. And then again, this is a long-term version of that, where over days you're seeing this reduction in the behavior elicited by the stimulus. And now, when we compare within a session how the animal responds to the novel orientation versus the familiar orientation, you can see that there's significantly more behavior for the novel. So this is a clear readout of memory retrieval, the readout being the novelty detection. But because we're recording in the mouse here and we're recording in the brain of the mouse, we can actually see that the cortical response is being modified as the animal learns. So here you can see a very pronounced form of plasticity, where the visual cortical response increases dramatically, the opposite of what we might expect actually, but increases very robustly and significantly over those same days. And again, we get that beautiful stimulus selectivity, this case with a larger response to the familiar stimulus than the novel, reflecting some kind of memory process within the cortex. And this is true whether we measure a very translatable signal like an event-related potential that we could potentially also record eventually in humans, or the kind of recording that's very difficult to do in humans, where we can record the activity of a single neuron. So looking at action potentials here, you can see that even the peak firing rate of individual neurons is increased by familiarity. So because we're working in the mouse now, we have the opportunity to use genetic tools to intervene in key circuitry, targeting key molecular mechanisms of this plasticity to really unpick what's going on in the brain. And so here you can see one example of this from our work, where we've targeted a particular cell type and a particular molecular mechanism that we know to be involved in synaptic plasticity. And this is the NMDA, a subclass of glutamate receptor. So we know that this is critically important for this process of habituation and the accompanying plasticity. What we're able to do now is target a genetic reduction, or in fact a knockdown of that NMDA receptor, only in a specific subclass of cortical cell. And this is the NTSR1 expressing layer 6 neuron. Okay, so you can see here that when we knock out the NMDA receptor only in these cells of the cortex, is there a dramatic reduction in this stimulus selective potentiation that accompanies habituation. And these layer 6 cells are very interesting. We're focusing a lot of our research on them at the moment because they provide the major feedback from the neocortex, in this case primary visual cortex, to the thalamus, which is a major relay station for information coming from, in this case, the visual epithelium, the retina, into the brain, transferring visual information into the cortex. And so this is an enormous feedback system. But they also have very interesting connectivity within the cortex, seemingly quite exclusively contacting within the region that we're recording these parvalbumin expressing inhibitory GABAergic neurons that Gemma Modinos mentioned earlier on. And we can also, just as another example of the kind of interventional approach that we take in mice, we can use optogenetics or chemogenetics, which are relatively new tools that can be used to activate or inactivate these specific cells on command. So we can use genetic tools to express an actuator, a particular receptor, only within those cells of interest. And then we can either use a drug to trigger those receptors or shine light on those cells and produce an inactivation in the case of the chemogenetics or an activation in the case of this optogenetics. And you can see that when we do that for these parvalbumin expressing cells, we completely remove the capacity of the visual cortex to discriminate familiar and novel stimuli. And that's whether we inactivate those cells, so we disinhibit the cortex and produce an enormous cortical response, or we activate those cells and inhibit the cortex to dampen down the cortical response. And so just to finish up, I'd like to just say that the other thing that's very interesting about habituation as a particular sort of fundamental foundational form of cognition is that it is deficient in a lot of psychiatric and neurological disorders. And so you can see here, you can create mouse models for highly penetrant genetic causes for instance, of neurodevelopmental disorders. And you can see here knockout of MECP2, which is the genetic risk factor that causes Rett syndrome, is a very debilitating neurodevelopmental disorder that manifests as intellectual disability. You can see here that the type of plasticity that I've introduced to you is severely deficient, and we know that this is reflective of a specific deficiency within those parvalbumin expressing inhibitory neurons. Likewise, at the other end of the lifespan, we could also try to model dementia. So here's an example here where a mouse model of tauopathy, which is a cause of several different forms of dementia. You can see here again, there's a deficiency in the stimulus selective cortical plasticity. And finally, even in normal aging, so where an animal is not in any way a model for a disorder, but we can track the aging of that animal, we can see that the amount of this plasticity, which can be measured repeatedly through the animal's lifetime with different stimulus sets, actually decreases as the animals get older. You can see that here in the latter stages of the animal's life, there is almost none of this plasticity. Interestingly, that cortical plasticity can be rejuvenated by application, chronic application of the antidepressant fluoxetine. And so this is, perhaps an indication again of a rejuvenation of some of those juvenile plasticity mechanisms, probably, which include the engagement of those parvalbumin expressing inhibitory neurons. And you can hopefully see where we're going with this. The next stage is to try and bring all of this back to the human. So we think that here at King's, there's a wonderful opportunity to translate this back into normal humans, but also patient populations, to try and use this plasticity as a readout of the function of the cortex. And as I've said, some of the measures that we use here, for instance, event-related potentials, could very easily be translated into non-invasive recording technologies. Okay, thank you very much for your attention.
Thank you very much, Sam. Beautifully intricate work. We are running over time. I do apologise, but hopefully you'll agree with me that it's been fantastic, and we've got a few more fantastic speakers to come. We're delighted to welcome another 3-minute thesis finalist. So Lucy Chester is an NIHR Maudsley BRC student. Are you ready, Lucy?
Yep, I'm here. Am I sharing the slide?
Great. You're on.
Is it sharing?
Yep.
Oh, perfect. Okay, I can't see it. What do you think of when you hear the word cannabis? And what does a cannabis user look like to you? Recently, you might have seen in the news a different sort of picture being painted. A picture of a young person smoking what they think is a harmless plant, only to find themselves believing and seeing things that can't possibly be real. Psychotic disorders, like schizophrenia, might only affect about 1% of the population overall, but they have such a profound effect on those who do suffer that researchers have made it a priority to find out what is causing these symptoms. And cannabis is often cited as a prime suspect. Now, most people who use cannabis, which is about 30% of the UK, will never get a psychotic disorder. But what we know is that people who do have a psychotic disorder are more likely to be cannabis users than those who don't have one. So the question now is, what came first, the psychosis or the cannabis? Well, one of the best ways of answering that question is obviously to look at what people do before they develop psychosis. Now, only about 1% of the general population develop psychosis over their lifetime, and 1% isn't a very big number to work with. So our research team went around interviewing people across the globe who are at ultra high risk of psychosis. What ultra high risk means is that because of their family history or some minor symptoms that they'd shown before, we could estimate that about 20% of our population would develop a psychotic disorder over 2 years. And looking at the bottom part of the slide, of the 344 people who we interviewed, 65 did. Now we want to look at what might have tipped those 65 over the edge. I want to look at cannabis use. So, were participants who had used cannabis more likely to get psychosis than those who hadn't? The short answer is no. And maybe that's our final answer. But that's just whether people had ever used cannabis. And after all, smoking 1 or 2 cigarettes isn't likely to give you cancer. So now we want to look at how much cannabis people are using. How often are they using it? And how old were they when they started using it? Remember that young people's brains are still growing and changing. So it might be that those who start using cannabis very young will see more of an effect than someone who's used the same amount, but started when they were older. Another thing we'll compare is potency. Like how whiskey has more alcohol in it than beer, certain kinds of cannabis contain more of a substance called THC. It's THC that gets you high, and potentially it's THC that might also make you psychotic. Studies have shown that cannabis growers are breeding their plants stronger and stronger to keep up with market demands, and this might be putting people like our study population at greater risk. Around the world, the laws surrounding cannabis are changing rapidly. The best decisions we can make when it comes to cannabis are those that reduce the overall risk of harm. But first, we have to understand where the harm really lies.
Great. Oops. Great. Thank you so much, Lucy. It's brilliant. So our next speaker is Dr. June Brown. June's ready, and I think, Trevor, you have her slides. Thank you. You can unmute, June, and you're ready to go. Thank you.
Can you hear me? Right. Okay. Thank you. Thank you, Mittal. I want to talk about accessible early interventions for depression and anxiety across the lifespan. Given that this conference is about the lifespan, I want to talk to you about different age groups that we've worked with. And I'm just very interested in early interventions, and especially accessible early interventions. So can we have the next slide, please? So why do we need early interventions? And the first reason is that over 75% of mental health problems develop by age 24. So this is what Tammy Moffat was talking about, and this is what Kessler in 2007 found. It's a very consistent kind of finding. So early interventions are therefore very important, whether in terms of actually focusing on younger people or whether in terms of reaching older people as early as we can, because there's problems of the persistence of problems and so forth. But, and it's a big but, only 30% with mental health problems will seek help from services. And so 70% will not be accessing help. And so a lot of the clinically effective treatments that we develop are therefore not used because people are not coming forward. So this is a kind of big issue that I would like to talk about. So common barriers to seeking help include stigma, how problems are perceived, and low mental health literacy. I'm sure there's a whole load of others, but those are the key ones. However, interventions which have usually attempted to change help seeking have only changed attitudes, and that was a review by Gulliver, and not behavior. So what we have done is we've developed a new model, and I'd like to tell you about this new model. So I'd be very interested in your views about this. So the model is based on a review by Gask in 2012 and mechanisms that we've tested in evidence-based one-day psychoeducational CBT interventions. So they're basically one-day CBT interventions. And we've used focus groups, we've used interviews, we've used RCTs to test the mechanisms. And we've developed this model, which is called the knapsack bridge model of help-seeking behavior. Why knapsack? It's to do with the letters that will relate to the different mechanisms, but it's also about hopefully a kind of nimble model that can be used in different settings with different interventions. And it's very much about help-seeking behavior. It makes a difference as to whether or not people do actually come forward. Right, can we have the next slide, please? So what is the knapsack bridge model? And there are two main parts to it. So it combines a non-stigmatizing pathway, which is at the top here, and a more acceptable intervention. And the two do need to go together. You might have a good pathway, But if the intervention is not acceptable, then it may not go very far. Or if you have an acceptable intervention, but without a pathway, then it may not give you what you want in terms of increased help seeking. So what are the letters and what do they stand for? So N is a normalizing title versus a diagnostic title. So I've talked to you about this numerous times, I'm sure. We used to run depression workshops. We used to run one-day depression workshops. And what we found was that very few people came. But when we changed the title to self-confidence, the difference was dramatic. Huge numbers of people actually came. So the title is really important, and it's about a normalizing title. The second E is about perceived effectiveness versus known effects. It's really to do with the fact that a lot of people don't really know how effective mental health treatments are. It links to the mental health literacy issue. But we need to somehow tell people that mental health treatments do actually work. P stands for publicity versus low awareness of services. Quite a lot of the studies talk about people who say, well, I didn't actually know about these services. So it's important to publicize the services that are being offered. And S is self-referral versus GP referral. So we have used a model where people can actually come forward by referring themselves. So they might see the service on a social media platform or through publicity at the GP service, or it might be word of mouth. But it's something about just saying people need to know about services in a way other than necessarily just going to their GPs to be formally referred. The IAP service does thankfully use self-referral, and a lot of people are using that way now. And it's a particularly important one in relation to Black and ethnic minorities, but we'll come to that in a bit. The other bit of this is about an acceptable intervention. The intervention needs to be acceptable. And there are interventions which have had problems in terms of acceptability. So some of the non-guided remote interventions have had problems in terms of acceptability and take-up. And the final one, which is C, is convenient versus inconvenient locations and times. We've tried to run interventions at the weekend or in the evenings or at times which would suit people. So we've asked them what would suit. Some of the answers have come back in a surprising way, but sometimes people will say, oh, I'd like to actually receive the intervention during the day as opposed to the weekend. So it's about asking people what would actually work for them. And so often we're trapped by the 9 to 5, Monday to Friday kind of mentality, because that's when we work. So it's something about just saying we need to be consulting people and asking them what would suit them. Right, next slide, please. So who have we reached? Using this model, we've reached a number of different populations. We've reached a general population of all ages who are depressed. So we've reached the 18-year-olds to the 70-year-olds, people who are depressed. That was Horrell in 2014. We've reached Black and ethnic minority groups, 30% often. So they are a representative percentage of people. We've reached depressed people in the local workplace. So this was when I said, you know, should we ask people whether they'd like to come at the weekend? But no, what they wanted to do was come during the working week so that they could access the intervention at work time. We've reached postnatally depressed mothers. So Van Liesholt is a Canadian psychiatrist who is using this approach with his postnatally depressed mothers. We've also reached 16 to 18-year-old students in schools. So we have done a feasibility study and reached 16 to 18-year-old students in schools. And young people are actually quite reluctant to seek formal help often, but we have actually reached them in the schools. And we have also reached male university students. So Sagar Uriagli has developed a pathway to male university students, and it has come up with some surprising results in terms of who male university students will actually go to and what kinds of people will actually come forward. So all sorts of interesting questions as a result of the model, but as I say, we've reached quite a number of different groups using that kind of model. Right, next slide, please. So where next? As I said, this is a new model, and it can be used in different settings with different interventions. So if you'd like to come to me to talk about it, please feel free. But in terms of where we are, we've now just got funding from the NIHR, HTA for 1.7 million. So we're running a 4-year trial. It's in 60 schools with 900 16 to 18-year-old students in London, Northampton, Manchester, and Bath. So COVID has obviously had an effect, but we're starting that up. It's called the BEST study with 2 S's, Brief Educational Workshops for Secondary Schools trial. It started in January 2020 and it's finishing in December 23. The intervention comprises day-long psychoeducational stress workshops called Discover, and these have been shown to engage Black and ethnic minority students, many of whom have not consulted before. We will, sorry, there's a typo here, we will assess clinical outcomes, health economic benefits, as well as do a qualitative study. And if successful, these workshops could be rolled out to all schools across the UK after the trial. So thank you very much for listening. And if you want to ask me any questions, please do that in the Q&A.
Thank you, June. It's very, very smart work. I'm going to hand it over now to Dr. Lauren Carson, who is a postdoctoral research project manager. And here we go. Are you ready, Lauren?
Yes. Can everyone see my slides? That's the first thing.
Yeah. And we can hear you too.
Excellent. Well, that's even better. So I will try and keep this brief. I realise it's gone over time a little bit today, and this is slightly different. So rather than presenting research findings, I'm actually here to demonstrate the wonderful ELIXIR partnership that I've had the privilege to work on over the last couple of years. So, as I said, I'll be giving a brief introduction to the Early Life Cross-Linkage and Research, or ELIXIR partnership, which is a collaboration across King's Health partners and is supported by the UKRI. This partnership is exploring the associations between physical and mental health problems between mothers and children through the use of de-identified electronic patient records and administrative data, with the potential to become one of the largest mother-infant-child datasets in Europe. So the ELIXIR partnership is a dynamic linkage using electronic patient records to create a dataset, as I said, of maternal and child healthcare data, providing population level life course data across South London. This data uses this dataset will use de-identified electronic patient records to provide potentially transformative information for life course research, integrating both physical and mental health disorders and outcomes in routine clinical care. The ELIXIR database has the potential to grow by up to 14,000 new maternity cases annually, in addition to providing child follow-up data. The physical and mental health care data for ELIXIR comes from 2 acute NHS trusts, which are Guy's and St Thomas's NHS Trust and King's College Hospital, and 1 mental health trust that we all know very well, which is SLAM. And it's collecting data from all patients in maternity and neonatal services. ELIXIR has the scope to allow for research across a wide range of areas, including women's health, infant and child health, healthcare management, mental health, public health and implementation sciences. The ELIXIR partnership operates an opt-out consent model and has been running since October 2018, which means that we do actually have some really interesting data that is hopefully going to be published later this year, looking at COVID as well. So, as I said, we've been running since October 2018, and in our data set we have over 36,000 pregnancies, and we have data on over 15,500 births across GSTT and KCH. In addition, we are also collecting data from neonatal intensive care units across both GSTT and KCH, and have data on over 2,000 neonates who've required intensive care treatment. At the moment, we have over 35 research projects underway using this data set, and the numbers of this are growing all of the time. And our data is refreshed every 6 months, and there are lots of new linkages that are currently pending and underway to expand this data set. The cohort profile was published in the BMJ Open back in October 2020. And presents the early descriptive data for the linkage database and the robust data security and governance structures, as well as the described intended expansion of the database from its original development. This image, although a little bit busy, shows the current pending and potential linkages we have for ELIXIR. So if we focus in the middle block, ELIXIR currently contains maternity and neonatal data, as I said, from the electronic patient record system known as BadgerNet, and we collect our mental health data from the CRIS dataset, which is based out of the BRC nucleus at SLAM. And we also have primary care data coming in from Lambeth DataNet as well. We are if we look to the left-hand side of this screen, we are currently in the process of incorporating national and linked hospital episode statistics data, which will include admitted patient, outpatient, A&E and adult critical care data sets. We're hoping to also include health visitor data via the electronic Red Book or e-Red Book, and artificial reproductive technology data, including IVF data from the Human Fertility and Embryology Authority. As we go over to the right-hand side of this image now, we also look to, in future, incorporate education data from the National Pupil Database, additional medication data from the National Prescribing Dataset, pollution data and family justice administrative data from the Children and Family Court Advisory Service, also support service, also known as CAFCAS. If you have any further questions about ELIXIR or would like to use the data, please feel free to contact myself or the Chief Investigator, who is Professor Lucilla Poston. And I'd like to thank you for listening, and also to the organisers for allowing me to present this today. Thank you.
Thank you, Lauren. That's brilliant. So we have our final speaker now, who's Dr. Petra Ploitzy, and she's currently on maternity leave, but has kindly pre-recorded a talk for us, which will be
Good afternoon, everybody. My name is Petra Ploitzy, and I'm an Alzheimer's Research UK Senior Fellow based at the Department of Basic and Clinical Neuroscience. I'm very happy to be here today, and in the next few minutes I will talk to you a little bit about my work on the role of blood metabolites in midlife cognitive function and Alzheimer's disease. So why am I interested in Alzheimer's disease? We know that Alzheimer's disease is the most common form of dementia, and it's a complex heterogeneous disorder with no disease-modifying treatments. So this highlights the urgency for the implementation of precision medicine approaches for the enhanced risk screening, detection and prevention, and ultimately treatment of Alzheimer's disease. So what we need is the right drug targeting a process upstream, given at the right time, at the right dose, at the right person based on their biological profile. And this is where blood biomarkers come. Deconstructing Alzheimer's disease risk into multiple genetic and biological subsets can provide us with an effective precision medicine biomarker-guided strategy. And in the last year, there's been a breakthrough in Alzheimer's disease biomarkers. So in addition to the well-established imaging and CSF biomarkers, there's been a number of blood biomarkers that have emerged. And these are easily accessible, minimally invasive, and scalable. However, there are still questions to be answered. So through my work, I try to answer some of these questions by using biomarkers and big data. So I'm using biomarkers in order to understand more about the biochemical pathways involved in Alzheimer's disease. What is the relationship of these biomarkers with lifestyle and environmental variables? How do they correlate with other omics layers, and can you detect them early? And very importantly, are they on the causal pathway and therefore to Alzheimer's disease and therefore amenable to intervention? And to answer these questions, I predominantly work with a specific type of biomarker. These are blood metabolites. These are small molecules such as lipids and vitamins, and they present the interaction between the genotype and the environment, and therefore both heritable and modifiable. And they sit very near the phenotype, so they provide us with a snapshot of our health status. They're also very relevant in Alzheimer's disease, as they can cross the blood-brain barrier, and they're also biologically plausible because the brain is highly enriched in lipids, and lipid genes, a number of lipid genes are involved in Alzheimer's disease. So how do we do this? So what we do in more detail is that we look at different phenotypes, such as clinical Alzheimer's disease, as well as cognition, cognitive function in midlife, and change in cognitive function, as well as brain atrophy and CSF and established blood biomarkers. We're working at the intersection between biomarker discovery, trying to identify these molecules and these pathways, and causal inference, try to see whether they are in the causal pathway to Alzheimer's disease and therefore whether they're amenable to intervention. We further integrate this with other omics modalities to try to understand more about the biochemical pathways involved. And finally, we try to investigate the relationship with lifestyle and environment through the life course. So, for example, our student Becky has used network and pathway analysis to try to identify clusters of metabolites and key drivers of these clusters, which are associated with late midlife cognitive function and change in cognitive function in the MRC46 birth cohort, as well as with brain atrophy in a subset of the participants. And this was followed by the exploration of the influence of life course factors from early life to middle life on these relationships. We have additionally looked into causal inference by using a method called Mendelian randomization, which is akin to a clinical trial, and our student Jody linked molecules such as HDL subfractions and glycoprotein acetyls, a marker of inflammation, which were previously associated with midlife cognition, again in the MRC46 cohort. We linked them to be potential on the causal pathway to Alzheimer's disease. Further, Jody expanded her work by looking at a wider selection of metabolites and whether they are on the causal pathway to Alzheimer's disease and whether they mediate the association of established risk factors such as education and cognition with Alzheimer's disease. So to this end, she identified again a subfraction of HDL, free cholesterol in very large HDLs, and glutamine to be potentially on the causal pathway to Alzheimer's disease, but these molecules were independent. They did not mediate the relationship of education and cognition to Alzheimer's disease. So to conclude, blood metabolites are good markers of cognitive function, and they are causal candidates for Alzheimer's disease. They can highlight new etiological pathways, and they can ultimately inform future treatment strategies. And I would like to thank you so much for your attention, and apologies for speaking so fast. But I try to fit a lot in 5 minutes. Thank you very much.
That's great. That's great. Thank you, Petra. So I'm going to hand over to Professor Matthew Grubb, who is our Vice Dean, also the Director of the BRC. So Matthew provided and will also thank you very much. Are you ready? Have we lost Matthew? I might need your help to add Matthew back. Okay, while we're waiting. He's a message. While we're waiting for Matthew, just take this opportunity with our speakers again. We had two really excellent speakers.
Mitchell, I'm back.
Great. Over to you, Matthew. Thank you.
Thank you very much. I've got a horrendous echo, which is slightly distracting. But thanks everyone for that amazing roundup of presentations. The annual research festival is one of the highlights of the Institute calendar, I think, and we had a series of presentations today which really covered so much ground across the IoPPN. The theme was across the lifespan, but it could also be said that we actually showed research across the career span. And actually it's one of the great pleasures of turning up to these, to hear presentations from PhD students all the way to senior professors, and hearing such diversity of work. So it's also across the disciplines, and we heard fundamental neuroscience to health services research and public policy. An amazing rich array of different types of research methodologies. And I think one of the things I enjoy most about IoPPN is having that kind of diversity, all focused around the nature of the disorders which we study, and the underlying mechanisms which contribute to those disorders. And of course, we could say that this was across disorders. We had everything from autism to age-related hearing loss, and many of the problems which afflict us. And we were reminded by Temi of how all of us are vulnerable to these disorders from across the lifespan. And finally, of course, across geographies, because not only are many of the samples we talked about international, but many of our speakers, of course, are international. And again, a wonderful, rich range of speakers working in many different continents. We heard about work in Africa, in New Zealand, and in the States, as well as closer to home in Europe. So I'm pleased to thank the contributors to this, all of the speakers, of course, to Mittel and Paolo, who did such tremendous work in chairing, even though the platform sometimes makes it more difficult to keep to time, and a number of people, and I hope I capture all or most of them, and apologies if I don't, but a number of people who've been involved in the organisation. Jonathan Omertashit, Marija Petrinovich, Josephine Mumford, Robin McGinn, Annika Ancliffe, Hannah Warren, Trevor Brooks, and as I say, I hope I've captured everyone, but if I haven't, please forgive me. Now, the problem with COVID, of course, is that we aren't face-to-face. We aren't all in the Wilson Lecture Theatre. We would, at this point, be having a wonderful round of applause for everyone. We would also be inviting you up for drinks in the canteen, which, of course, we can't do now. But I'd like you all to sort of take the opportunity, I guess, to raise a glass metaphorically to the work which has been going on, and thanks again everyone who's contributed to this wonderful research effort. And I close the meeting.